Selamat pagi, salam sejahtera. Good morning. Yang berbahagia, Datuk Dr. Chu, Vice President of Minds and Chairman of the Organizing Committee. Yang berbahagia, Professor Engineer Dr. Leong, Vice President of International Network of Women Engineers and Scientists and Module 2 Chairperson. Yang berbahagia, Datuk-Datuk, Datin, Professor, Yang Mulia Tengku, distinguished speakers, girls, young girls, researchers, all distinguished participants, ladies and gentlemen. Welcome to MINDS 2020 National Conference on the contribution to national development by women, Malaysian women scientists and technologies. Now, this is the third international conference organized by MINDS in conjunction with the National International Women's Day, which falls on the 8th of March. The UN theme for the International Women's Day 2022 is gender equality today for sustainable tomorrow. Now, although this equality is yet to be achieved, but I'd like to highlight the work of some outstanding women scientists, in particular, Madam Curie, who has won 2 Nobel Prizes in scientific field. No human being has achieved this feat. Also, Dr. Rosalind Franklin, who contributed significantly to the double helix structure, and unfortunately she missed the Nobel Prize due unto unforeseen circumstances. But King's College of London has named a building after her in appreciation of the work she has done. Now, coming to home ground, I would like to congratulate, to highlight the scientific work of Professor Dr. Miao, winner of the Outstanding Women Scientist Lifetime Scientific Research Achievement Award, and also Associate Professor Dr. Tai. We also have the benefit of the presentation by other eminent Malaysian scientists. We are convinced that this program will inspire young girls not only to pursue their education and career in science, but also to be leaders and innovators. Finally, I would like to make some acknowledgement. The success of this conference due to a number of people. First, must congratulate again Professor Yao and Associate Professor Dr. Chai, to thank IET, Institute of Engineering and Technology, for the sponsorship. The last but not least, our appreciation and gratitude to Yamabaga Dato' Chu and her team for their commitment and dedication to make this conference a great success. Thank you.
Thank you, Tan Sri Ong, for your kind words. Good morning, Yang berbahagia, Academician, Emeritus Professor Tan Sri Dato' Augustine Ong Sun Hock, President MINDS, Yang berusaha Professor Dr. Ngiao, Professor Dr. Leong, Dr. Ravika Devi, honored guests, distinguished speakers, participants, MINDS Council members, ladies and gentlemen. I'm honored to chair the keynote address at session 1 of the conference today. The keynote address will be delivered by none other than Professor Dr. Ngiao Yun Fong, the recipient of the MINDS 2022 Outstanding Women Scientist Lifetime Scientific Research Achievement Award. Prof. Ngiao is an internationally renowned medical microbiologist. She is a medical doctor by training, a teacher, and an administrator who has contributed much to community and our nation. Without much ado, it is my pleasure to invite Dr. Ravika Devi, Minds Council member to do the citation for Professor Niao. Dr. Ravi, the platform is yours.
Thank you, Dr. Chu. A very good morning, ladies and gentlemen. Many women in science have devoted their professional lives to teaching, practicing, and promoting their respective fields of expertise. They have made tremendous strides, and their work will endure. Thus, to be singled out as a recipient of the Minds Outstanding Women Scientist Lifetime Scientific Research Achievement Award is an incredible honor. Previous recipients of this prestigious award were Professor Datuk Paduka Khatijah Yusof and Datuk Ambika Srinivasan. It is thus an honor for me today, on behalf of Minds, to give the citation for the recipient of the third Minds Lifetime Achievement Award, Professor Dr. Niao Yun Fong, for her decades of work and giant contribution to understanding and managing the smallest creatures on the planet, that is, microbes. Prof. Niao, a fellow of the Academy of Sciences Malaysia, graduated from the King Edward VII School of Medicine in Singapore and went on to get a master's in medical microbiology from the London School of Hygiene and Tropical Medicine. She became a member of the Royal College of Pathologists in 1984 and Fellow of the Royal College of Pathologists in 1996. She served on the faculty at 3 different universities or colleges as both a teacher and an administrator, namely University of Malaya, Unity College International, and University Tunku Abdul Rahman, or Utah for short. She was appointed Professor of Medical Microbiology at the University of Malaya Medical Center, where she was the Chief of the Hospital Infection Control Unit and Head of STD Laboratory. She is currently a Senior Professor at the Department of Preclinical Sciences, Faculty of Medicine and Health Sciences, Utah. She is also the Chairperson of the Center for Research on Communicable Diseases, Utah, and Consultant Microbiologist to Asunta Hospital, PJ. In her career spanning over 4 decades immersed in understanding microbes, Professor Niao established a strong international presence. She has served in various leadership positions, including as panel member of the World Health Organization Scientific and Ethical Review Group, Assistant Secretary General, International Union Against Sexually Transmitted Infections, Vice Chairman, International Union Against Sexually Transmissible Infections, Asia Pacific Branch, and Editor-in-Chief of the Asia Pacific Venereologist, just to name a few. Professor Niao does not stop. She is currently working on establishing research collaborations with the Chinese Center for Disease Control and Prevention and the Chinese Anti-TB Association. She has a strong academic background. Her research on bacterial pathogens, their genomic characteristics, resistance to antibiotics, and clinical relevance have led to numerous publications, including 9 book chapters, 3 laboratory manuals, and 166 publications. One of her recent publications was entitled Genomic Diversity of SARS-CoV-2 in Malaysia. Prof. Niao and team sequenced SARS-CoV-2 respiratory isolates from the National Public Health Laboratory of Malaysia and showed that the virus accumulated an abundance of mutations in the first year of the COVID-19 pandemic in Malaysia. However, the general genetic diversity was low relative to other Asian countries with larger populations. Professor Niao takes great pain and pride in inspiring and mentoring students as she believes in igniting young minds to create the future. She has supervised 14 PhD, 35 MSc, MD or MPath, 36 Diploma BS MB candidates, and mentored 10 WHO fellows. She is also highly innovative. As evidence of her innovativeness and enthusiasm, Prof. Niao led the team that clinched the first prize at the Sing Health Duke NUS Institute of Medical Simulations, or SIMS Challenge 2020, Innovative Application of Game Technologies. This was a virtual international event showcasing games for healthcare, training, and education. How did she become such an inspirational teacher and mentor? Who sparked her mind during her formative years? Well, the apple doesn't fall far from the tree. Professor Niao's father was the late Professor Niao Zichan, a prominent Chinese physician based in Kuala Lumpur during the mid to late 20th century. He made tremendous contribution in the field of traditional Chinese medicine and was known as the father of modern traditional Chinese medicine. He worked hard to champion traditional Chinese medicine in Malaysia and the region, to inspire the cultivation of the future generations of professional and responsible practitioners of traditional Chinese medicine. Ladies and gentlemen, I present to you the daughter of the father of traditional Chinese medicine, Professor Dr. Niao Yun Fong. Thank you.
Thank you, Dr. Ravi. Ladies and gentlemen, I'm sure all of you have been waiting for this moment and are as eager as I am to listen to the keynote address. It is with great pleasure that I welcome Prof. Niao to deliver her appreciation speech and keynote address.
Yang Bahagia Academician Emeritus Professor Tan Sri Datuk Dr. Augustine Ong, President, Malaysian Invention and Design Society, Datuk Dr. Chew Guan Mei, Vice President, Malaysian Invention and Design Society, ladies and gentlemen. I'm deeply honored and grateful for this award given in recognition of my lifelong quest to understand microorganisms that can cause human disease. I thank Professor Yu Hontat, President of Utah, for nominating me for this award. Professor Emeritus Chua Hian Tik, for bringing me into the Utah community with whom I spent some of the best years of my academic life, both socially and professionally. And I thank my colleagues and students in the University of Malaya and Utah for their enthusiastic and unfailing support in my research undertakings. This award is for all of us. When I embarked on my journey in academia after graduation from medical school, I had no great ambitions. I just wanted a job so that I could be financially independent. When I joined the medical microbiology department in the University of Malaya, I knew next to nothing of microorganisms and what they can do to us. But the training I received and my subsequent work experience gave me a lifelong fascination with the little creatures that are not even visible to our naked eye. I learned to fear them for their ability to cause disease, but I also learned to respect them for their ability to survive despite desperate human efforts to get rid of them. Working with microbes is never easy, but always a learning and growing experience. It is so true that the more you seek, the more you'll find. And the more you know, the more you realize you don't know. Every research project was a challenge, and every scientific publication an encouragement to do more and better. I have truly enjoyed my journey in science, and I hope my award will inspire at least some of you to create your own path to self-fulfillment. In closing, I would like to say to all the young scientists in our midst this morning, believe in yourself. And when you get an opportunity to achieve something worthwhile for yourself and for society, go for it. Thank you very much. Good morning. In this talk, I would like to share with you my attempts to better understand microbial pathogens that are the cause of infectious diseases in humans. Microbes are an inherent part of life on this planet. They coexist with humans, animals, and plants, and can be found in the air we breathe in, the food we eat, and the water we drink. We even inherit them from our parents and pass them on to our children, as scientists have found nucleic acids of viruses in human sperm cells and ova. Many of the microbes are beneficial, or at least not harmful to us. Unfortunately, there are some that can maim or kill us. These are the pathogens. The presence is indicated by a tissue response from the host. On the skin, host response is usually seen as red swellings that feel warm and are painful to touch. In other parts of the body, there may be indications of loss of function. In infected patients, we expect to see pus cells in lesions, increased white blood cells, as well as antibodies and sensitized lymphocytes in the patient's blood. In silent infections, the patient may not show any external signs. The course of infection may be acute, when signs and symptoms appear early and last only for a short period, or chronic, when signs persist for a long period, or recurrent, when signs appear on and off over a long time. Pathogens are called primary pathogens if they regularly cause disease in healthy individuals. They are opportunistic pathogens when they cause disease only in people who are already weakened by either disease or medical treatment. The lesions caused by pathogens can be localized to certain tissues or organs, or systemic when the infection is widespread with multi-organ involvement. In the 16th century, an Italian physician, Fracastoro, proposed that tiny particles, which he called spores, could transmit infection by people touching contaminated surfaces or breathing in contaminated air. But the germ theory, which says that illnesses can be caused by germs, was only fully developed in the 19th century, mainly by other European scientists, Semmelweis in Hungary, Robert Koch in Germany, Louis Pasteur in France, and Joseph Lister in the United Kingdom. These scientists collectively proved the bacterial causes of infections and demonstrated the efficacy of sanitation and vaccination as preventive measures. In Asia, we have our own great physician scientist, Dr. Wu Liante from Penang, who controlled the pneumonic plague epidemic in northeast China in the latter half of the 19th century. He demonstrated for the first time the person-to-person transfer of the bacterium that caused the plague and ended the epidemic that killed more than 600,000 people by stopping transportation out of affected areas, burning corpses to kill the plague bacillus, and implementing the wearing of masks to prevent the spread of infection. The wool mask he invented is the forerunner of the N95 respirator we use nowadays for high-level protection against airborne pathogens. The institution of preventive measures based on the germ theory was so successful that by the mid-20th century, many experts thought that infectious diseases were going to disappear altogether. Sadly, till today, we are still learning to cope with dangerous and deadly pathogens which continue to emerge and show resistance to antimicrobial drugs. This slide shows the major viral epidemics since the last century. For us, the 1999 Nipah virus outbreak was the most sensational, a new infection causing almost 40% mortality among humans, the culling of about 1 million pigs, and almost total collapse of the pig rearing industry in the country. Till today, Nipah encephalitis is still being reported from across Asia, where there have been at least 16 outbreaks in India, Bangladesh, Philippines, Cambodia, Thailand, and Laos, with mortalities up to 70% in some populations. And now, of course, we're struggling with COVID-19. This pandemic is a good example of how a minuscule microbe can bring us, the mighty humans, to our knees, devastating not only our health, but our socio-economic system as well. Among bacterial pathogens, especially, and fungal pathogens as well, the main problem is the continuous development of multi-drug resistance that threatens to end the usefulness of antimicrobial therapy. This multi-drug resistance pandemic is less dramatic than the viral pandemics and has drawn less attention, but is nevertheless a grave problem to be tackled before microbial superbugs get totally out of hand. It's clear that we humans need to regain an upper hand to prevent our own extermination. And we can only do this if we know our enemy well. As the famous Chinese strategist Sun Tzu said in his book, The Art of War, if you know the enemy and know yourself, you need not fear the result of 100 battles. So why are pathogens so difficult to deal with? For one, the microbes have the advantage of numbers because they can multiply very fast and can produce a new generation in just minutes or hours compared to an average of 20 years in humans. During their replication, changes can occur in their genetic makeup. These genetic changes can make them look and behave differently and help them to adapt better to their environments to survive adverse conditions. Using the COVID-19 analogy again, you can see how new variants are selected out successively under the pressure of acquired immunity from a natural infection or COVID-19 vaccination. Pathogens need to invade and multiply within the host. They do this by producing proteins called virulence factors, which help them to attach onto a surface, gain entry into a host, spread within the host by either escaping from host defenses or destroying host defenses. The host responds by trying to capture the invading pathogens and either neutralizing them with antibodies or killing them off with sensitized lymphocytes. If the host defenses win, the pathogen is eliminated. If not, the host suffers damages. Sometimes overly active host defenses can get out of control and destroy their own tissues along with the pathogens. A good example is again the cytokine storm in COVID-19, where host pathogen interaction produces excessive amounts of cytokines, which are small proteins that control the activity of host immune cells. And it is this cytokine storm that causes the severe widespread tissue damage, leading to multi-organ failure and death. This knowledge is very important as it tells us that in the treatment of infections, just targeting the pathogen is often not enough. We also need to be aware of the host reaction to infections to help us select the best therapy for our infected patients. So how can we get to know the pathogen better? When I started as a microbiology diagnostician a few decades ago, I had only microscopy, culture, and simple biochemical tests to identify microbes in samples from infected patients. Then, a technique called polymerase chain reaction, or PCR, was introduced in the 1980s. This method for amplifying DNA sequences offered a completely new approach to the laboratory diagnosis of infectious diseases. Without the need for culture, it became possible to detect and identify the microbes in patient specimens within a few hours. The host genetic predisposition to disease can also be determined. This was a major game changer that opened the door to the application of many other varieties of molecular diagnostic tests in clinical laboratories. For this remarkable innovation, the American inventor Kary Mullis received the 1993 Nobel Prize in Chemistry. Another important advancement is the use of whole genome sequencing for diagnostic and public health investigations. The genome is a complete set of DNA containing all genetic information of an organism. Genome sequencing is the method used to determine the entire genetic makeup of an organism. With whole genome sequencing, we get information useful for microbial identification, classification, and evolution studies, and for the study of pathogen virulence, adaptation, antimicrobial resistances, etc., etc. Practically everything you wish to know about the organism. Although whole genome sequencing is still largely a research tool, whole genome sequence-based assays have been introduced into the routine diagnostic service and is becoming more widely used as it becomes more affordable. One important use of genomic information is microbial genotyping or fingerprinting. As microbial genomes undergo changes like mutations, gene gain, gene loss, or gene rearrangements, each microbe will have its own genetic profile. Strains with similar genetic profiles are clustered together as genotypes, and it's believed that all strains in the genotype have a common origin. This fingerprinting technique is very useful for strain differentiation and has become an important tool for tracing the dissemination of microbes, for instance, in the investigation of outbreaks. The distribution of genotypes helps to answer questions such as, is this a novel infection? Is this a new transmission? Where is the reservoir of the pathogen? How is it spreading locally, nationally, and internationally? What is driving the spread of infection? And how can the outbreak be controlled? To make significant observations, we often need to examine a lot of data. But it's not easy to gather a lot of data by our own efforts. This is where in silico analysis has become useful. These in-computer analyses make use of data deposited by scientists all over the world into public databases that can be accessed online. The most often used public database is GenBank, established in 1982 by the National Center for Biotechnology Information, or NCBI, in the United States National Institutes of Health. This open access database provides annotated collection of all publicly available nucleotide sequences, as well as their protein translations. GSAID, or Global Initiative on Sharing All Influenza Data, was established in 2008 to provide genomic data of influenza viruses. But since January 2020, it has also been providing data on SARS-CoV-2 and has been very useful for the comparison of SARS-CoV-2 genomes, assessment of local and global transmission, development of vaccines and diagnostic tests, and surveillance on the emergence of new virus variants. This much-quoted SARS phylogenetic network figure from Foster et al., published in the Proceedings of the National Academy of Science, USA, illustrates the type of information that can be generated with worldwide genome data. It shows the genomic relationship between the bat coronavirus, RATG13, and the SARS coronavirus 2 viruses isolated from patients in East Asia, Europe, and USA. Interestingly, RATG13, which was suspected to be the cause of the early infections in Wuhan, showed a closer relationship with SARS-CoV-2 viruses circulating in Europe and the USA. Recent publications from a group of French researchers also suggested that the pandemic SARS-CoV-2 virus is likely to have originated not from RATG13, but from bats in Southeast Asia. In Malaysia, Utah collaborated with the National Public Health Laboratory, MKK, and Dr. Tan Jun Liang in Multimedia University to publish our own observations on the genetic diversity of the SARS-CoV-2 virus in the country from January to December 2020. This work was also facilitated by our access to genome data deposited in GSAID by other Malaysian researchers. So we come to my endeavors in the past few decades. I'll illustrate how I've used some of these modern tools in my own studies. In my early years working in a hospital diagnostic laboratory, my main concern was how to detect bacterial pathogens that were either difficult to grow or took too long to grow in the laboratory. So DNA amplification tests based on polymerase chain reaction and ligase chain reaction became the chosen tools for the diagnosis of infections by the atypical pathogens, chlamydia, mycoplasma, and Legionella. These bacteria are difficult to grow. Quite often, the patient's signs and symptoms are not specific for any infection, and the laboratory has to put up multiple tests for all possible infections in the same patient specimen. To reduce workload and shorten reporting time, multiplex PCRs were used for the simultaneous detection of likely pathogens in specimens. For instance, for genital infections, a multiplex PCR assay was developed to look for the 3 common urogenital pathogens, Neisseria gonorrhoeae, Chlamydia trachomatis, and Ureaplasma urolyticum. And for respiratory infections, another multiplex PCR was developed to detect the 3 common respiratory pathogens, Mycoplasma pneumoniae, Chlamydia pneumoniae, and Legionella pneumophila. For bacterial typing, I started with simple serotyping and bacteriocin typing, which were phased out when more powerful genotyping methods were introduced, such as the PFGE, MLSD, RAT-PCR, PCR-RFFP, and VNTR typing for various pathogens. The most productive work was the typing of the TB bacillus, which I'll describe later in this presentation. My work with whole genome sequencing had an easy start, as the beginning of the 21st century was the time for rapid publications of draft genomes. Draft genomes are incomplete genomes. This practice was actually encouraged by well-established journals, and I guess there was a great interest then in the scientific community to gather as much genomic information as quickly as possible. My colleagues and I published more than 20 draft genomes in just a few years on mostly non-tuberculous mycobacteria and fungi. More extensive genomic analyses were later done on specific microbes of interest. I was fortunate to be involved in the genomic characterization of Malaysia's first extremely drug-resistant TB bacillus, XDR-TB. This work was headed by the late Professor Dr. Ng Ki Ping at University Malaya. He also shared with me his prolific publications on his collection of dermatiaceous fungi, wood-decaying fungi, and allergy-eliciting fungi recovered from human patients. With Dr. Chuk Jagdi and Professor Rosmawati Muhammad in the University Malaya, we reported on hepatitis B and hepatitis C variants in infected patients. My work with antibiotic resistance mechanisms was focused on the use of genetic and genomic investigations on resistance mechanisms affecting important rescue or last resort drugs for multi-drug resistant infections. These antimicrobials included carbapenems, colistin, tigecycline, linezolid, and the drugs for the treatment of extremely drug-resistant tuberculosis, which are bedaquiline and clofazimine. Genome analysis provided all possible resistance determinants, such as cell membrane changes that may decrease antibiotic intake, efflux pumps that may increase antibiotic extrusion, and enzymes that can either degrade antibiotics or protect antibiotic targets. We need to know how bacteria can become resistant to antibiotics so that we can design novel, effective antibiotics that will not be affected by any existing resistance mechanism. Again, I was fortunate to have a chance to work with Dr. Hao Wang from Ningxia University in China. Dr. Wang and his colleagues were synthesizing inhibitors of the enzyme dihydrofolate reductase as novel antimicrobial drugs. And I tested the synthetic drugs against the TB bacillus, as well as the few common multi-drug resistant bacterial pathogens to provide a biological validation for the in silico molecular modeling experiments. The bulk of my genomic studies were on the mycobacteria. This slide shows the classification of mycobacteria into Mycobacterium tuberculosis, which is the cause of TB, Mycobacterium leprae, the cause of leprosy, and a large group of non-tuberculous mycobacterial species, including Mycobacterium abscessus, which is the species I worked with most in my research. Mycobacterium abscessus is notorious for its resistance to many antibiotics. Besides antibiotic resistance studies, my students also investigated its evolution and subspecies classification. Dr. Tan Jun Liang, currently with the Multimedia University, contributed strong evidence for the separation of the subspecies Macillians from the subspecies Bolletii with his phylogenomic network analysis, as shown in this picture. We also described the genomes of two potentially novel non-tuberculous mycobacterial species related to Mycobacterium iranicum and the Mycobacterium terri complex. One of my most enjoyable projects was a study of tuberculosis among elephants in 6 elephant sanctuaries in Malaysia. This was a study initiated by Dr. Ong Bee Lee from UPM with the assistance of the Wildlife and National Parks Department and the Ministry of Health. We examined elephant trunk wash for the presence of Mycobacterium tuberculosis and took blood from the elephants and their handlers for TB serology. The results showed a higher seropositivity among elephant handlers compared to the general public, and evidence of both active and latent tuberculosis among the elephants. These results suggested frequent, close contact and a two-way transmission between the elephants and the handlers. From the trunk wash of elephants, we also isolated 2 non-tuberculous mycobacteria, which appeared to be identical phenotypes. So we examined them with a whole battery of tests, including conventional biochemistry, phenotype microarray analysis, protein profiling with liquid chromatography, mass spectrometry, PCR sequencing, repetitive sequence-based PCR typing, and whole genome sequencing for phylogenomic analysis. The results of all these tests showed them to be possibly novel, slow-growing NTMs with highly similar biological and genotypic characteristics that qualified them to be classified into the same NTM species. We were also informed that the 2 elephants were handled in the same sanctuary 8 years apart. Thus, it is possible that both elephants were exposed to the same source of infection in that shared sanctuary. My most productive research project has to be the genotyping of TB isolates from Malaysia. With the fingerprinting method called spoligotyping, my students and I were able to identify major clusters of the TB bacillus in the country and compare them to known clusters in other parts of the world, map the distribution within the country, and determine the amount of active transmission of TB in different parts of the country. Our main conclusions were that there's a predominance of the Beijing clade in Malaysia, which reflects our predominantly Asian origin, but there's also a widespread occurrence of East African Indian strains, which reflects the influx of populations from other parts of the world. In addition, the occurrence of the ST19 clade, which is common in Manila, and the CAS clade, which is common in Pakistan, suggests substantial intra-regional population movement. This is the CITVIT web, which is a map of all Mycobacterium tuberculosis genotypes determined by scientists in different parts of the world, giving a global view of MTB genetic diversity and associated demographic and clinical information. It allows us to pinpoint circulating lineages of the Mycobacterium tuberculosis, to draw conclusions on evolutionary trends and TB geoepidemiology. I'm proud to say that Malaysian scientists have contributed more than 2,000 polygotypes to this global map. So I've come to the end of my presentation. Before I leave, I'd like to say that all the advancements in science and technology have given us only a glimpse of the complexity of the microbial world and how it is intertwined with human and animal existence. It is obvious that we still need a lot more information to know how to reduce microbial pathogenicity so that humans and microbes can co-evolve in peace. For my work, I owe my thanks to many local and international collaborators who have provided funding and technical assistance, and especially to my colleagues and students in local universities, without whom I would not have been able to achieve any of the scientific accomplishments in my career. Thank you all for your attention.
Thank you, Professor Niao, for your excellent talk on understanding microbes for better disease control and showing us the power of microbes even though they are very small creatures. Ladies and gentlemen, since this is a keynote paper, there will not be a Q&A session. Congratulations again, Prof. Niao, for winning the MINDS 2022 Outstanding Women Scientist Lifetime Scientific Research Achievement Award. Ladies and gentlemen, we will now commence session 1 of the conference. There are 3 papers in this session to be presented by eminent scientists covering the topics of agriculture, marine biology, and communication in science. Each speaker will be given 20 minutes, and all Q&A will be taken together at the end of session 1. Our first speaker of session 1 is Dr. Sim Sun Liang. Dr. Sim is currently resource person with the Sarawak Biodiversity Center. She is a member of the Technical Committee on Protection of New Plant Variety. She has received several honors from Sarawak State Government for her contributions in science and community. She is a fellow of the Academy of Sciences Malaysia. She is a fellow in the Academician and Expert Workstation of the Spices and Beverage Crops Research Institute under the Chinese Academy of Tropical Agricultural Sciences, PR China. Please welcome Dr. Sim Sun Liang. She will speak on plant breeder, a creative and fulfilling profession.
Okay, good morning, Academician Tan Sri Emeritus Professor Dr. Augustine Ong, President of MIND, Malaysian Invention and Design Society, Dr. Chu Yan Mei, Deputy President of MIND, eminent academicians, MIND Secretariat, students, distinguished guests, ladies and gentlemen. Thank you, MIND, for this opportunity to speak at the National Conference on Contribution to National Development by Malaysian Women Scientists in conjunction with International Women's Day 2022. Indeed, It is a great privilege to be part of this event. Today, I will be talking on a topic, plant breeder, a creative and fulfilling profession. There you see me there. Plant breeder is an applied geneticist who undertake plant breeding. And plant breeding explores natural existing variability. It uses hybridization and genetic engineering techniques to develop new plant varieties with desirable characters. Conventional plant breeding technique included crossbreeding, which involved combining two sexually compatible species to create a variety with the desired trait of the parent. The steps involved in conventional breeding are 6: collection of germplasm, evaluation and selection of parents, crossing among the selected parents, lab and field evaluation over multiple generations to select the best offspring, multi-location trials, and multiplication of the improved plants. Mutagenesis is another plant breeding technique which uses mutagen such as radiation and chemical to induce random mutation, creating the desirable trait, and is most successful in floriculture plants. Polyploidy breeding is another breeding technique. In this case, the example here shows the seedless watermelons are created by crossing a plant with 2 sets of chromosomes with another one that has 4 sets of chromosomes. The offspring, which has 3 sets of chromosomes, is sterile, that is seedless. Now we have new plant breeding techniques, including genetic engineering, which enable addition of genes from any species, DNA marker assisted selection, which can shorten time for breeding process, and genome editing, which at the moment is considered the most efficient and targeted modification of plants. These techniques increase the effectiveness and efficiencies of plant breeding. Genetic engineering is addition of genes from any species or knock down a gene to produce new variety with desirable trait. Example here is the rainbow papaya, which is modified with a gene that gives it resistance to papaya ring spot virus. This slide shows the selected trait modified by genome editing. The latest new variety of crop developed by new plant breeding techniques are golden rice with increase of beta carotene, which the body can convert into vitamin A, and GABA tomato contains higher levels of an amino acid, which can believed to add relaxation and help lower blood pressure with 1 or 2 tomatoes per day. Plant breeding has made enormous contribution to global agriculture in yield resistant to bio and abiotic stresses, improve of quality trait, and long storage period for harvested crops. Now I'd like to talk a bit on my work on pepper. Malaysia is one of the major producers of pepper in the world. 90% of pepper is cultivated in Sarawak, and it is the most important cash crop for smallholders who mostly living in the remote area of Sarawak. And the crop is suitable for them because the products can be stored. Pepper is known as king of spices. These are some products of pepper. Pepper cultivation faces many problems. The major one included pests and diseases, and also a labor-intensive harvesting because pepper flower is not synchronous and the fruit ripening is uneven. It needs many rounds of manual selective harvesting to complete harvesting of the crop. So it is desired to breed for pepper that is high yielding, resistant to major pests and diseases, and synchronous in flowering. When I first start my pepper breeding work, I faced so many constraints. The main one is a lack of gene pool, limited gene pool, lack of any variety that is resistant to major pests and diseases, and lack of research facility. I was given only 2 hectares of secondary jungle to start with. No plant houses, no laboratory. And there was no simple and reliable method of artificial pollination for pepper. So I went on to study the floral biology of pepper and mode of pollination of pepper. And I found that pepper is mainly self-pollinated, and the pollen is descended either by gravitational force or distributed with the help of dew or light shower or wind carried spray or rain. The stigma appeared before the anthers, so we can make use of these characters to devise our breeding methods. So this is the new breeding method that I developed, not new, but improved. So the work is carried out in a plant house instead of in the open field, and using this stage to do the artificial pollination, we don't need emasculation. And the flower spike is not wrapped in poly bag like my predecessor has done, because if you use polythene bag, I found sting built inside the bag and rainwater also collected inside the bag. So the spike is no longer viable. So instead, I use nylon shears material as the isolation bag. And the pollination is done by dispensing pollen suspension into the stigma. So with these methods, I carry on pepper breeding work. And this slide shows the development of Smongo amas, a new variety of pepper which takes 20 years to develop. Starting from 1971, I propagate and planted the progenies, which was from crosses done by my predecessors. I carry out a 3 replicated trial, do a few inoculation, and selected the one that is resistant to the disease. But this hybrid is not high yielding, so I have to do the back crossing to the high yielding parents and start the breeding cycle again. Until 1984, I start conducting a field observation over 138 foot rot disease infested pepper farm in Sarawak. Finally, in 1981, we have new variety developed. This new variety is less susceptible to major diseases, Phytophthora, foot rot, and blackberry disease, and less susceptible to pest, pepper weevil. It's more even in even ripening, so it reduces the round of harvest, cut down the cost of harvesting. Its yield is comparable to high-yielding traditional variety. There's another new variety developed during my service. It's a selection from germplasm collected from Costa Rica. Its yield is comparable to the high-yielding variety. This variety is very pungent, is hot, because it has higher content of oral resin and piperine. And it's also quite even in fruit ripening, less susceptible to foot rot disease. Furthermore, the branch spike can be exploited further for developing high-yielding varieties. In addition to these 2 new varieties, I also generated some 30,000 of hybrid seedlings and open pollinated progenies, which enable further selection. This slide shows the 3 potential new variety of pepper. During my trip to collect germplasm in South America, I also collected Arachis pintoi, a leguminous plant to be used as cover crop to control soil erosion. But I also found there's a lot of landscaping company use it for landscaping. So I would say the following is my gratifying achievement. First, the release of 2 new recommended variety of pepper and their wide adoption by pepper growers in Sarawak. The introduction of Arachis pintoi as cover crop and for erosion control. And I expanded the gene pool of pepper, generated over 30,000 hybrids and open pollinated progenies for selection. Plant breeding is mundane and with little glamour. However, I find it is a creative and fulfilling profession. Creative because you design the type of plant which you wish to develop. Fulfilling because it gives you a sense of pride and satisfaction each time you achieve success in providing growers better planting materials. Plant breeding is both an art and science. Art because you are designing the plants. Science because you need knowledge of genetics, plant physiology, agronomy, reproductive biology, statistics, and molecular biology technology. A plant breeder needs strong determination, diligence, and perseverance. On career opportunity, I would say a person who is trained in plant breeding might find position in a university or government and working in corporation in the plant breeding and seed industry. In the words of Nobel Peace Prize winner Norman Borlaug, plant breeders have made an enormous contribution to food production, global agriculture, and the general well-being of mankind and have a tremendous potential to do so. So I'm not hesitant to recommend younger generation of scientists to take up plant breeding. You now have access to the new plant breeding techniques, which enable improvement of plant to be faster, more precise, more targeted, and more exciting. It can be regarded as rewarding as you can obtain plant breeders' right and collect royalties. It can be fulfilling when you see the fruits of your labor being recognized and passed on as your legacy for the good of mankind. Thank you.
Thank you, Dr. Sim, for your excellent presentation. Our next speaker is Professor Dr. Eileen Tan. She's a professor in School of Biological Sciences, University Science Malaysia. She's a fellow of Academy Sciences of Malaysia. Currently, she's a director of Center for Marine and Coastal Studies in USM. She was the first woman president of the 59-year-old Unitas Monarchologica, which is based in Belgium from 2013 to 2016. In 2020, she was awarded the Top Research Scientist Award in Malaysia for expertise in mariculture and marine ecology, biodiversity, and conservation. She is the best woman scientist for her work in guiding the women folk in Johor Islands in earning their own income through the giant clam conservation program. She will speak on science impacting life through sustainable aquaculture. Professor Dr. Eileen, the floor is yours.
Good morning, good afternoon, and a good day to everyone, wherever you are. First of all, I wish to thank the organizer of Malaysian Invention and Design Society, MINDS, for giving me the opportunity to share on how I translated my science and research findings to the community. The title of my talk is Science Impacting Life Through Sustainable Aquaculture, with the tagline of Going Green with Mollusk Culture. Ivory towers are places where knowledge, discoveries, inventions, and innovations are generated. Ivory towers are where great minds discuss and finding solutions to solve issues, be it local or global issues. But how many of this knowledge, innovations, discoveries from research have been translated to the communities, to the bottom billion, and making impactful solutions and differences? For all I know is most of these findings or results or discoveries stays in the publications on the publication in the library and stays within the ivory tower. Today, I would like to share with all of you about using science and research findings to eradicate poverty and to ensure food security through green aquaculture. The reduction in captive fisheries and resources due to over-harvesting, degradation of habitat, and climate change had caused more than 90% of our local traditional fishermen receiving monthly income of less than 500 ringgit per month per household. We need to move forward to aquaculture, not just any aquaculture, but green aquaculture. Why do we need to go green or move towards green aquaculture? Green aquaculture can ensure Malaysia's food security, environmental protection, sustainable future, and more profitable in the long run. There is a high demand for food and living space increases, leading towards alternative modes of food production. We are also looking at a more health-conscious society, looking towards consuming healthy food, and green products are more acceptable now. One may ask, why oyster farming? Why not prawn or fish farming? So let's discuss about this. Let me share with all of you on the advantages of oyster aquaculture. There is a high demand for oyster locally in Malaysia itself. Malaysia is currently only producing 14% of the oysters consumed, with 68% of the oysters are imported. The imported oysters can be in the form of live fresh oysters, frozen, canned, dried, or even in other forms. Oyster farming technologies are relatively easy to practice compared to prawn farming or fish farming. The research findings have been simplified and modularized into smaller sections that can be readily adapted and applied by the community. Oyster farming is also low in labor costs and low in its operational costs. It is low in operational costs because the oysters feed on natural phytoplankton that is naturally existing in the culture site, which is really readily available. The maintenance of oysters are also low, with no feeding schedule required, and that's very low in terms of disease. University Science Malaysia has started conducting research into oyster biology, breeding, and farming since 1988, which is 30 years ago. And UC Science Malaysia has proven technologies to support this green aquaculture. On top of the advantages, oyster produced from Malaysia is definitely fresh and delicious compared to those frozen imported oysters. Even the live flown in imported oysters are not as fresh due to being out of the water during the long transportation hours. Many people have this misconception of oysters. Oysters is a shellfish, therefore is high in cholesterol. But that conception is wrong. Oysters, in fact, are a very healthy food where it is super low in cholesterol. Eating 1 dozen of fresh oysters is actually providing only as much cholesterol as a quarter of an egg yolk. Oyster has been recommended as a healthy protein source for heart patients in the Western countries. Of course, Malaysian oysters are internationally acceptable and palatable. And oysters has always been known for its aphrodisiac value. Malaysian local oysters are actually far better because it is super fresh, almost as fresh as directly been harvested from the farm compared to imported oysters, which many times are flaccid and watery. Malaysian oysters are of course cheaper and can taste much better due to its freshness, and research findings have developed oysters with the local oysters with better taste, better texture, and also better shape to be served as half-shell market, both in restaurants and in hotels. Oyster farming is clean because the whole farming does not require antibiotics. No feeding, since its feeding is on the natural phytoplankton. When there's no feeding, it does not pollute the water with excessive feed. No cutting of mangrove, which makes oysters an ideal candidate to protect the environment, yet able to fetch a good price. Oysters are able to boost the local economy. Center for Marine and Coastal Study, known as CIMEX, from University Science Malaysia, has started conducting research on oysters since year 1988. And CIMEX has successfully joined hands with the industry to build the first commercial oyster hatchery in Malaysia. In fact, it is the first commercial oyster hatchery in the region. Well done, Malaysia. With science, research and innovations, USM, CIMEX is able to produce oysters of better quality and able to grow fast compared to other oyster producing countries like Australia, New Zealand, USA, Canada, and Japan. Through research and science innovation, USM managed to close the breeding cycle in the lab, in the laboratory, as well as in the field, which is the grow out. We are able to culture the oysters from day 1 until 12 months and ready to be harvested and to be marketed. How do we translate the science from the laboratory to the community? What we did at the very early stage or initial stage was to listen and to understand the needs of the community. We must understand their culture, beliefs, and also their lifestyle of the community. We must make sure that the science that we want to share is what is needed by the community, and the community is able to learn and to adapt into their life, into their lifestyle. It's not about us going to the community and tell them what to do without first understanding what they need. We must also show trust, respect, able to listen to them. And most of all, the scientists need to provide strength and empowerment to the community. We must create confidence in them so they are willing to take up the challenge to do new things. It is not about forcing the community to do what we think is best for them, but we must play a role to ensure that the community understand the opportunity and the science and knowledge that the scientists are going to share with them. In oyster farming, science was translated to the community part by part. The science of 30 years research has been simplified and been compartmentalized in order for the community to adapt the whole procedure. In other words, the size of oyster farming has been converted into biteable sizes that can be chewed and digested by the community. Something easy and simple enough for the community to do on a daily basis and able to continue doing in a sustainable manner. We have translated the complicated science findings into simple portion that can be conducted by the community. With this approach, the community feels more comfortable in learning new things and are more willing to be committed to the farming activities. They started off working on a part-time basis because the community still needs to gain an income to sustain their livelihood. At the early stage, the community will still continue their fishing, farming, and others, where they will work in the oyster farm after their daily work. They do it as on a part-time basis. The scientists work closely and hand in hand with the community through a process we call co-learning, co-production, and co-creating to find solutions and creating new approaches on the ground. Soon the scientists and the community are working as a team with full respect and trust among both parties. It is about a team working as one. This is not about the scientists translating or wanting their science to make to be used, but it's about translating the science to make impactful difference in the community. We translated the knowledge of science and skill to make oyster farming a success for the community, upgrading their livelihood. We taught them how to build the floating raft for the oysters, the science of monitoring survival and growth, how to protect the oysters from predators, and also the importance of taking care of the environment to ensure that the area is still good and fertile for oyster farming in a sustainable way. The researchers have also successfully bridged the gap between the industry, government, and academia and community through oyster farming, and also at the same time taking care of the environment. We always believe in the concept of giving a man a fish, we feed him for a day. But if we teach him how to fish, we feed him for life. We have proven through oyster farming that science can make changes in the life of others. Maybe not to many, but at least to Pak Su and his communities. The smile in their faces tell us all on what differences science has made in their lives. Before science entered their lives, their monthly income is only around 350 to 700 ringgit per month. But with science coming in and using science, their income have increased to more than 3,000 ringgit per month. We are celebrating our impacts made by science to the community. Pak Su and his community are now social entrepreneurs. They have utilized oyster farming to practice conservation and tourism through green agriculture. Pak Su, science has changed Pak Su from a person who doesn't speak a word of English to now, where he can brief the whole process of oyster farming to foreigners and to use these students, to visitors, to tourists in simple English. Besides being green aquaculture, oyster farming is also a sustainable aquaculture where the activities can be conducted successfully without destroying the mangrove or the environment. Even the community knows how to explain. They understand the importance to create a balance between income generating through oyster farming and to protect the environment for sustainable aquaculture. The science The science of oyster farming is providing impacts not only to the farmers, but also to the women folks and children of the area, creating more opportunities of improving their livelihood, education, and welfare to all. The women folks can now generate income by cooking for tourists, visitors, students, instead of just staying at home as housewives. Even the children who once has no pocket money when they go to school, now able to have at least 2 ringgit as their daily allowance to school given by their parents who involved in oyster farming. 2 ringgit might not sound or seems to be a lot to most of us, but to them, it is a different. It is a different from none to 2 ringgit. Oyster farming is also a result of science addressing and fulfilling Malaysia's international obligation, such as Sustainable Development Goals, namely SDG number 1, which is no poverty, SDG 5, gender equality, SDG 8, decent work and economic growth, SDG 13, climate change, and most of all, SDG 14, life below the water. The journey of educating and making a change in someone's life is a long journey and a challenging one, and is filled with many, many challenges. It requires commitment from all, not only just the scientists, but also from the commitment from the community. So the take-home message, last but not least, the take-home message to all the scientists and researchers out there who's listening to this talk, the most useful asset of a person is not a head full of knowledge, but a heart full of love, with ears open to listen and hands willing to help. With that, I end my talk, and thank you everyone for your kind attention, and stay safe, everyone. Thank you. Thank you, everyone. Stay safe. Bye-bye.
Thank you, Professor Datuk Dr. Irene, for your excellent and insightful presentation. Our final speaker for session 1 of the conference is Datin Technologist, Dr. Mahalakshmi. She's Executive Director, Malaysian Biotechnology Information Center. She's a biotechnologist specializing in science communication, and is the founder of Petri Dish, the first scientific newspaper in Malaysia. In 2015, she received several awards, including 100 Most Influential Persons in Biotech in the World Award, Women in Biotech Law and Regulations, and Women of the Future. In 2010, she was awarded the World Academy of Sciences Regional Prize in Public Understanding in Science for Southeast Asia and East Asia. Today, she'll be talking about science communication for women innovators. Please welcome Datin Dr. Maha.
Good morning, everyone. I'm so happy to be speaking on science communication for women innovators. I think this is a very important topic because a lot of time scientists think that just doing research and publishing is their job. And here I strongly believe in what Sir Mark Walport said. Science isn't finished until it is communicated. And the communication to wider audience is part of the job of being a scientist. So how we communicate is absolutely vital. So I will be talking about the core duties of scientists and how this actually can be incorporated into science communication and how this science communication benefits scientists. So science communication actually starts with the minute we start to introduce ourselves or our research. And many times we introduce ourselves, right? To investors, to policymakers, to politicians, or collaborators, industry collaborators, or other researchers from other institutes. So the minute we start introducing ourselves, science communication has already started and you are communicating science. And how do we get attention through this introduction? How do we give the impression that we are going to be very relevant to the audience and we are not boring, and we can build trust, credibility, and show the impact of our work? So by giving a strong introduction using science communication principles, we can achieve this. So I'm going to introduce this template, 2793 template. So what it means is, every time we want to relay a message to our audience, our key stakeholders, always limit that to 3 messages. And these 3 messages, each message use 27 words. And for each message, use 9 seconds. So that is 2793. So here I prepared something that's going to introduce my talk. So this is my introduction. Science is very powerful. It provides socio-economic and environmental well-being. How do we bridge the disconnect between science and society? How does science communication benefit scientists? So that is what I'm going to talk to you today. So if you look at my introduction, the last sentence, benefits scientists. I made it very relevant to today's audience, which are scientists. Of course, I can talk about how science communication benefits policymakers, farmers, or investors, but I'm not going to touch that. So my focus is on scientists. So this is my introduction. So think about an introduction that will create an impact whenever you tell people who are you and what research you do. And that is already science communication. So why communicate science? Now, I believe before a scientist decides that I want to communicate science and become passionate about science communication, scientists must be inspired to communicate science. And I feel as a communicator, this is still lacking among many scientists, not just Malaysia, but many other countries as well. So let's see why we need to communicate science. We need policies and regulations that are science-based. Scientists are doing excellent research in the laboratories. But for that research to go into the market and farms and the public, policies and regulations must support the research. And we see a lot of regulations stifle research. Now, economic growth. I always say there is no one country in the world that has attained developed nations without a strong pillars of science, technology, innovation. So we as scientists, we need to communicate science. So science, technology, innovation becomes strong pillars of the nation. Now we also see a lot of time, Ministry of Science is really a small ministry with small funding. How can we make that into a bigger ministry? So this is where science has to be communicated. And another reason is citizen participation. We also see that a lot of time citizens are not involved in science. Science belongs to the elite, like researchers, STEM professionals, but we need citizens to set the priority, tell the policymakers, set the pressure for policymakers to put priorities on science. And we also see non-acceptance from among the public new researchers like vaccines. There was so much of problems in accepting vaccines. So we need citizen participation. Another thing is we need science to become culture and lifestyle, not just economic and politics, environments and sports to be the talk of the town, but we want science to be the culture. Talk about science at dinner table at home. Talk about science at kopitiam and have science cafes, musicals and centers where it can be a leisure time, platforms for families to spend time. And we need to communicate science for science to be commercialized, because commercialization involves non-technical audience like TTO, technology transfer officers, investors, banks, and policymakers. So we need to communicate science. So now scientists will say that, oh yeah, everything looks so good. It is, I see the benefits, but how does it benefit me as a scientist? Because I am a scientist, I'm not trained in science communication. I don't have the resources, funding, and human power. I am busy with teaching, publications, and consultation, admin work, and I have so I'm busy with all these things, research. So how can I communicate science? And why should I communicate science? If we look at all the benefits of science communication, it actually brings the benefits back to the scientists. So industry collaboration. For industry collaboration, we need to speak the industry language, the dollar and cents language, the public acceptance language. And we all know a lot of science that happens in the public sector research institutes and universities. Without industry support, it cannot come out to the market. So we need industry collaboration, and for that we need to communicate with them in their language, repackage our science, our research. As I said, regulations. Scientists are doing great job. Even in Malaysia, for example, scientists were doing great job in genetically modified crops, but many scientists stopped their work in this area because they found the regulations to be very stifling. So they cannot just do research and expect someone else to bring policies and regulations that are very friendly. So scientists have to communicate with the regulators, with the politicians, with the lawyers. So this is where science communication helps. As I said, funding. For commercialization, we need funding, and this funding does not come from someone who understands science. The policymakers in the ministries are not scientists. So how can we speak to them? We need to communicate science in a way that they understand. A lot of time, it's not just the ministries giving funding now, it is crowdfunding, angel funding, completely from people who do not understand science. We need to garner attention. commercialization, political support, public acceptance. Now, I want to introduce this word social licensing. It's not the licenses given by regulatory authorities, but the license given by the public. We saw how mRNA vaccine was feared by the public, rejected by the public in the beginning. So we need public acceptance, which is social licensing. And for that, we need to communicate science. And civic scientists, I'm a strong believer scientists has to be the idol, the celebrities in the forefront, role models for more women and girls to take up this field. So for all those reasons, scientists have to communicate, and you will see that these benefits actually come back to you. So let's look at the traditional way. Scientists do research and then they publish in journals. It's read by the peers. Of course, this is important for critics, advancement of research, and professional development. But how about doing the same thing, but add on popular science? Talk about your science in research in blogs, tweet about it, write popular articles in media. And if you do that, then it's not just going to reach the peers, but also other scientists, policymakers, politicians, farmers, investors, media, public. So it's going to have a wider reach. So this is the benefit of science communication. Very quickly, I'm going to talk about the bloopers by scientists. Many scientists are communicating, but we often do a lot of mistakes. And even science communicators like me, we are all trying, we try and error, we make mistakes, and then we learn. So very quickly, what are the common mistakes made by scientists? The single biggest mistake is scientists think that it has been communicated or someone else will communicate. We often think that, what is that the public doesn't know? Everything is in the media, they can Google. So it is there, communication is already taking place. No, there is still a lot of confusion, misconception, fear mongering. So we need to communicate science. So, and everyone has to do that. And a lot of times scientists get very excited because we have got so much of data and we are excited about our work, the impact, the applications, and we want to give everything to our audience. So too much of information is not good because it shuts off the audience. Remember, science is complicated, abstract, and the public will not understand this. So it confuses audience. And if we start our science communication journey with the wrong foot, we are going to lose our audience. Once we lose our audience, it's going to be very difficult to get them back. So not too much information. I always say our job is not to create another PhD through science communication. Our job is to give them enough information for them to make the right decisions, for them to get excited, just for them to sensitize so that they can start reading on their own and they know how to differentiate science and pseudoscience. Not giving the bigger picture. Now, scientists, all of y'all do a very niche, narrow area of research, in-depth research. So when we talk about our research, a lot of scientists just immediately zoom into your narrow research without giving the bigger picture. So people are not able to put that narrow research into perspective and into the bigger picture. How can they cannot take that piece of your research and put it into the big picture because it's very complicated and it's really very in-depth. So try to give the bigger picture. If you do apoptosis, for example, in cancer research, give the bigger picture of cancer and how apoptosis, your research on apoptosis is going to help in treatment of cancer. Trivializing science. Now, communicators like me always tell you, please make it very simple, not too much of depth in what we are doing, just like what I presented earlier. But having said so, I see some scientists, they completely dumb down the science. Now, this is also a mistake because it is science communication. We still need science in our stories, in our messages. And remember, science is intriguing. Science is fascinating to the public as well. So don't dumb it down until there's no science in our stories, in our messages. So make sure there is enough science to trigger the interest, the curiosity, and answers the questions, the concerns that the public have. So this is a very common message. Science communicators will tell you, KISS. Keep it short and simple. This is very important. But again, as I said, don't trivialize science. Make sure there is enough science to excite the audience. Now, in a real workshop, I will really go in depth into all these things and give you tips and hacks how to do it right. But I'm going to go very quickly just to sensitize you on how it can be done. And of course, you can also figure out through experiments. Now, another thing is no jargons. Now, let me also contradict myself here. We always say don't use jargons, but not too many jargons, but choose at least one or two jargons which are key signatures in your research, so that it becomes a daily vocabulary for the public. Now, we all know DNA, for example, is a jargon, but it became a vocabulary in English, in Malay, in normal conversation. We always say, oh, it's in your DNA. Football is in Malaysian DNA. Makan is in Malaysian DNA. How did it become a common vocabulary? And today, mRNA, PCR has all become a common vocabulary. How did that happen? Because we continuously started using mRNA. We continuously started using PCR. So that became a vocabulary, and people, they might not really understand it, but at least people are using it, and some of them have made their own research and they understand it. So these are how we can choose one or two jargons, keep repeating it and explain it, so it becomes a daily vocabulary, but not all the jargons in your research. It's too much for the public. There's no one size fits all in science communication. So it does not mean I tell you something and you can immediately cut and paste that in your research and communicate that in your research. That does not happen. Science communication has to be customized. When today, if I'm talking to scientists, it's a different language. Tomorrow, if I'm talking to students, completely different language, different messages. If I'm talking to farmers, different messages, different style, different languages. So the message is the same, but I repackage it so that it becomes relevant to my stakeholders, to my audience. So that is how we communicate science. So, it takes a lot of time to customize, and that is why my job just keeps going, because I cannot keep using the same messages, keep using the same articles for different audiences. But this makes science communication really interesting, because it's challenging to really change the same message, but how do you turn it around for different messages, different stakeholders? And not too much of how. Scientists are always into methodology. How do you do it? And we start even our communication on how. So not too much of how, because people want to know the why first. Why? First of all, why are you doing this? Why is it important? Why should government put money into this research? So the why is more important than the how. Remember, in a scientific paper, the how methodology comes before the impact that we have created. But in science communication, it is the how. So you see, start with the why. Why am I doing this research? Why is it important? Then slowly, when the public is triggered, they want to know more. Slowly we move on to how. How do I do this? And what is it? What is the product? What is the services? What is the research? So always start with why. So you see, when we start our conversation with why and then slowly move outwards, how and what, it is remarkable conversation. If we start from what, how, and finally why, it is conventional. It is very a conventional conversation, and it's very difficult to trigger public, especially when we are talking about something that's complicated, that's not relevant to them, or they don't see the relevance. So by talking about why, they immediately see the relevance. It creates empathy among our audience, and audience wants to know more, which gives us opportunity to now talk more about our research. So with that, thank you so much for the time. And I communicate science using all of my social media. So you can also see how I repackage my messages and make it relevant. And sometimes I also call for action. I want my audience to do something. So I want them to read more. I want them to ask questions. I want them to make a change in their lifestyle. So a call for action is important, and I do that in my social media as well. So you can also check out my social media on how I communicate science. And I think social media is really an important tool because we don't have to get permission from anyone. Of course, we stick to the guidelines of our institute, but then we don't have to have an editor who will say, no, your research is not interesting or it's not relevant to my readers. So it is our field, our platform. So I would urge scientists to slowly start communicating your research in social media. So with that, thank you so much.
Thank you, Dr. Maha, for your very interesting and insightful presentation on the importance of communicating science. Ladies and gentlemen, that concludes all the 3 presentations in session 1 of the conference. So now it's my pleasure to invite the 3 eminent speakers, Dr. Sim, Professor Dr. Irene, and Dr. Maha, to join me for the Q&A session. Dear participants, so all of you are here already? Yes. Okay, thank you. Now, dear participants, those who wish to ask questions, please type your question in the chat box, mentioning your name, organization, and the speaker to whom you wish to address. I have some questions here. Let me start with question 1. This question will be addressed to Dr. Sim. This is from Mr. Asman Ismail, Organization, Academy of Tropical Agricultural Sciences Association. The question is, as this is the conference on contribution to national development by Malaysian women scientists, would you like to tell us specifically your contribution? Dr. Sim, would you like to address this question? Thank you.
Thank you, Dr. Chu, and thank you for the question. I would say the development of the new varieties of pepper, which is more tolerant to major diseases and pests, which is comparable with the high-yielding traditional variety, and which is more even in ripening, which cut down the frequency of rounds of harvest, and also with higher quality, are considered my contribution to the pepper industry in the country. The new variety are now planted by, as estimated by the Malaysian Pepper Board, is planted by some 70% of the pepper holding in Sarawak. And it is also used by a famous spice company, McCormick. McCormick is establishing a plantation, a pepper plantation in Terengganu. And the company adopted our new variety of pepper for his plantation. So I think I have only this one single one, which I consider a contribution. But in addition, I think the fact that many of my former postgraduate students and research assistants who are now key researchers in the various institutions, particularly in Sarawak, can be considered as my contribution to human resource development for the country. Last, I want to mention my contribution to science. I think as one of the co-author of a publication of pepper genome, I consider this is a contribution to science. Thank you.
Thank you, Dr. Sim. We now move on to the second question. Question 2. Okay, this is by Miss Roshini, student Tunku Abdul Rahman, University College. The question is addressed to Dr. Eileen Tan. Question is, what is the biggest challenge that you had to face to ensure the success of your work with the community? Dr. Eileen?
Yes, thank you, Dr. Chu. Okay, thank you, Miss Loshini, for your question. The biggest challenge I had to face to gain acceptance is to gain acceptance from the community. Being a Chinese female marine scientist can be very challenging, especially when I need to work closely with mainly Malay male-dominated coastal communities. I have to show respect first instead of expecting to be respected. As a scientist, I cannot just enter a village and tell them, the villager, since I'm a professor, a doctor, to do this and to do that, and what is best for them. Working with the community is not as direct or easy as following any modules, as in the laboratory. We must first gain the trust of the community through listening, observing, and understanding. Trustworthiness is important to secure a long-term relationship between the scientists and the community. And of course, we have to have open mind when we are in the community. It's a whole process of co-learning and co-production of knowledge findings. It's amazing when we are in the community, we as a scientist, we learn a lot of indigenous knowledge from the communities themselves. That is, I feel, is the main challenge. As soon as we can open up ourselves and stay humble and be part of them, then it's already a breakthrough to working with the community. Thank you. Back to you, Dr. Chu.
Oh, thank you, Professor Dr. Irene. Okay, the next question is addressed to Tatin Technologist Dr. Maha. This is from Mr. Nathan, Primary Entertainment in Yampahat. The question is, how does communicating research help women scientists in particular?
Thank you so much. Yes, thank you so much, Mr. Nathan. Thank you, Dato. Now, I think this is an interesting question. I have explained this, but this question is particularly, how does it help women scientists? Now, women are often, we ourselves, we don't really network so much. The boys have got the boys club, and then they've got someone who knows from their school days, and their friend's friend is their friend. Women are very timid, and women also have this imposter syndrome. I've heard very prominent scientists in Malaysia who holds very high position, and even she tells me, not naming anyone, she tells me, I always feel, how come I'm invited for this? Am I that good? And she's really prominent, and often I have that too. So this is where communicating our research gives us the limelight and shows the impact of what we are doing to the stakeholders that I spoke about, to the investors, to our superior, that I need more grants. I need to expand my lab. I need more grants to recruit postdoc fellows. So for all these things, I think women, from the timid nature that we are, communicating research brings our research to prominence, and this gives us all the benefits that will help us to bring our research to commercialization. So that's a very short answer to what you asked.
Thank you. I think Dr. Maha. All right. We have the next question. This is a question to Professor Irene. This is from Dr. Tan Sui Lian from MINDS. Okay, the question is, you mentioned that oyster farming does not require feeding, as the oysters can depend on the naturally occurring phytoplankton. Now, how would you ensure that the phytoplankton population remains sufficient over many cycles of oyster farming? Professor Dr. Irene?
Okay, yeah, thank you, Dato. Thanks for the question, Dr. Tan Sui Lian. For every process, before we work with the community, we start up, we will go down the ground to do a survey on site selection, to choose the site with good water quality and rich with phytoplankton that is suitable for growth and survival of oysters. Not all areas are suitable for oyster culture. Some areas, they do have phytoplankton, but not the type of phytoplankton that is suitable for the oysters. And we also conduct carrying capacity of the areas to ensure we do not overburden the ecosystems, and ensuring sufficient sources of phytoplankton for the oyster farming. So in addition, also water in the rivers and mangrove areas are always flowing with ever available nutrients. The nutrients will be supporting the bloom of the phytoplankton and ensuring the phytoplankton is able to grow and multiple at all times to support the oyster farming. Yeah, thank you. Back to you, Dr. Chu.
Thank you, Dr. Prof. Yes. The next question, yes, this is also addressed to your good self, Professor, Professor Dr. Irene. This is from Dr. Valerie Aposa, Embassy of France to Malaysia. So the question is, do you consider there is enough support at national and international levels to develop this kind of approaches? Should there be more incentives for scientists to work towards community development rather than focusing on academic work and publications, etc.? Thank you very much for your talk. Prof. Dr. Irene?
Yeah, thank you, Dr. Chu. Thanks, Dr. Valerie, for your question. Well, in my personal opinion, there are available fundings from the Malaysian government and also some funds from international organizations to encourage scientists to work closely with the local communities. Lots of emphasis on the bottom billion, making changes in their livelihood. For example, Ministry of Higher Education provides translational research funds and community innovative funds open to all researchers to translate their research findings to the community. In the international arena is the UNDP small grant program that is also open up for all researchers to work pertaining to creating social impacts. And in the USD systems, we are now looking into creating a promotion track, not only on publications, but on the impact or on the efforts being put into sharing our findings with the community. So if so that is new incentive, besides the fund, there's new incentive for promotion among the younger scientists also. However, what being said, what is still lacking is maybe the contribution from the industrial sector, where most of their corporate social responsibilities, commonly known as the CSR contributions, are usually relatively short-term or one-off. It will be fantastic, if the corporate sectors can also join hands with the academia and the government to reach out to the community to make a difference and create a better tomorrow for all. Thank you, Dr. Valerie, for your questions again. Back to you, Dr. Chu.
Thank you, Prof. Dr. Irene. Okay, the next question is addressed to Tatin Dr. Maha. Is from Ida Itayu from UTM. The question is, for the effective innovation nowadays, which question should be a priority, what and or why you do it? Sometimes community accepts the science work very well, but not easily goes to scientists such as high impactors or vice versa. Is it mainly due to the communication skill? Tatin, Dr. Maha.
Thank you, Idayu, and thank you, Dato, again. Now, okay, I'm not a practicing scientist, although I'm trained as a scientist. There are so many prominent scientists in this room now, but I think still, for even being a scientist, the why question is very important. It's not what should I do when I do my PhD. It's not what should I do when I grow up. Even for children, I always tell parents, don't ask children what you want to be. Ask them why you want to be that. What is your contribution to the field? So I still feel, even in the field of innovation, why am I doing that? Sometimes we are not going to reinvent wheels. We are not going to completely start something new in our research. But what is already there? Why is it that way? How can I improve that cost effectively, sustainability? So I think the why question is really important. And this is where, again, caveat, I'm not a scientist, but for us, for our papers to be accepted in journals, now editors are also looking why this paper? What is the relevance? So if we can write in a way that our research, the why is very strong, I think the acceptability of papers, even in high impact journals, will be higher. This is where I've heard one workshop, an editor was giving a training on how to get our papers published. He was saying we can be doing research that's based in Malaysia, maybe a marine life in Terengganu, or floods in Terengganu, Kelantan, or Malaysia, or whatever. But then why is it important for in terms of global challenges? How do we relate that to global challenges? Now, that is what makes our paper relevant to any international journal, not just Malaysia, but relating it to, a focus on how in the bigger picture. So again, coming back to the bigger picture. So I think even communicating research for the scientific, among the scientific community, the why question and science communication principles will be very helpful. Thank you.
Thank you, Dr. Maha. The next question is also addressed to your good self. It's from Bahar Sakuna, ECA Automation Solution. Now, how to turn every new finding in science or technology into profit? Dr. Maha?
Okay, again, so many other scientists there. I'm not sure that if I'm the best person to answer this, but this is my personal feeling. I don't believe every science has to be profitable. I'm a strong advocate for basic research, fundamental research. And Dr. Eileen was saying translational research. We need all these things, not just applied research. Look at CRISPR-Cas. CRISPR-Cas is basic research, how the DNA system works in defense mechanism, and now it's being used for gene editing. So these are examples of, and CRISPR-Cas gene editing is used not just agriculture, but in every technology, industrial biotechnology, food technology, gene drives in every technology, but it comes from basic research. So when we are doing if you're doing basic research, you might not see how it's going to be applied, how it's going to turn into profit. And in fact, it doesn't have to turn into profit in your hands. But once it becomes successful, the concept is well established, someone else will take it up for profit making. And not just, I'm not talking about profit, but I like it to be seen as for the benefits of humankind. It doesn't have to be profit, but for the benefits of humankind. So don't worry too much of profit if you are doing basic research or even translational research. If you do it well, I really think it will become beneficial in many areas, socio-economic, environmental, and of course the economic what aspects are there. Thank you.
Thank you, Dr. Maha. I concur with you that both basic research and applied research are equally important. Thank you. Now, the next question is from VI Chin, Nature Farming Kuching. This is addressed to Dr. Irene Tan. Can you comment on risk of pollination over time, quality control, and certification, if any? Dr. Eileen?
Okay, thank you, Dr. Chu. Thank you, for your VI Ching for your question. Again, for your information, in our earlier site selection, before we actually venture into oyster farming with the community, we will usually look for areas away from development, away from industrial areas, to avoid risk of pollution, because areas, if let's say for example there is industrial areas upstream, we will avoid those areas because industrial areas will usually contribute to heavy metal pollutions. So those are the areas that is a no-no for oyster farming. We also try to educate the local farmers on quality control and bringing in the industrial partners to assist the communities to process the oysters to better quality oysters that are safe to eat, oysters of better quality, and following to that, consumers are more than willing to pay for better quality and safer oysters. And also, we are working closely with relevant departments such as the Health Ministry and also the Department of Fisheries on certification. At the moment, what is required is just a test report from any testing lab to ensure that there's no bacterial contamination of the oysters. But when we reach a stage that our oyster industry in Malaysia is ready for exporting our oysters, then more certifications are required. So it's all in the pipeline now. We are still in the midst of encouraging more participation from the communities to build the oyster industry big enough that we have enough to actually talking about export. Thank you. Back to you, Dr. Chu.
Thank you, Professor Dr. Irene. Now, in the interest of time, we'll just stop here for the Q&A. However, dear participants, if you have any questions, please send them to us and we will give you a written reply. All right. So, ladies and gentlemen, please join me once again to thank the three eminent speakers for their excellent presentation. Now, to all the participants, we would also like to thank you for your support and active participation. Ladies and gentlemen, we will now take about 10 minutes break before we commence session 2 of the conference. Now, the session 2 will be chaired by Professor Dr. Leong Wai Yi. She's the council members of MINDS and council members of IET. She's also the Vice President, International Network of Women Engineers and Scientists. We'll see you in about 10 minutes time, 11:05. Is that correct? Yeah. Okay, thank you. Thank you, everybody. Have a fruitful day. Thank you.
good morning. I'm Leung Wah Yi. In conjunction with the celebrations of the International Women's Day on 8th of March, Malaysian Invention and Design Society, MINDS, is pleased to present the third National Conference on Contributions to National Development by Malaysian Women Scientists and Inventors. This year, the year 2022 Young Scientist Award goes to Associate Professor Dr. Chai Lai Ching. Dr. Chai is a microbiologist and lecturer at University of Malaya. She's the chair of the Young Scientist Network Academy of Sciences Malaysia that represents top young scientists and researchers in the country who are committed to contribute to the national ecosystem beyond their individual research interests. Part of her role includes engaging young academics in the country to rethink their role in shaping higher education for the future. Dr. Chai's research focuses on infectious microorganisms, microbiological risk assessment, and microbiological food safety. Dr. Chai served as the Vice Chair of the Technical Working Group of Microbiology under the Food Analysis Committee, chaired by the Department of Chemistry Malaysia, and also is listed as the Malaysian expert on microbial risk assessment in the ASEAN Risk Assessor Directory. Also, she leads numerous academia industry projects, such as serving as key opinion leaders of 3M Asia Pacific, food safety research with Kikkoman Japan and other local food manufacturers. Her active contributions and involvement in microbiological safety research has won her the Malaspina International Award by the International Life Science Institute in 2017, and she is currently serving as the scientific advisor of a Southeast Asia region, providing guidance and advice on food safety risk assessment. Dr. Chai is also involved in promoting biosafety and biosecurity in Malaysia. Her research and leadership in promoting research integrity and excellence has been recognized through multiple awards, such as the L'Oreal UNESCO Women in Science Award 2018, the ASEAN Women Entrepreneurs Leadership Awards 2019. So let's welcome Dr. Chai. Congratulations again. Thank you.
I would like to express my utmost appreciation to MINDS for giving the Young Women Scientist Award 2022 to me. I was super happy and proud the minute I received this unique trophy from the delivery and hold it in my own hands. It really means so much to me for being recognized, empowered, and being supported. Actually, many has asked me this question: why is it so important to have a special award just for women scientists? Is it necessary? And of course, I would like to say that I would like to be just called as a scientist, not being addressed as woman scientist, as I believe that science has no gender, and it doesn't matter if you are women or men. Both genders would make good scientists as long as you work dedicatedly, passionately, and ethically in research. The need for more women in science goes beyond fairness and ethics. Although both women and science are of similar species, but they definitely do not build similarly. Having more women in science will actually bring different and unique perspectives to research and to scientific conversation. Having diversity, be it gender diversity or ethnicity diversity, or even disciplines of both art and sciences in the research ecosystem, will add to the collective intelligence for science advancement. Gender diversity and equality is very important, not just because of human rights, but also because, ensuring gender balance is important for building peaceful and a prosperous nation. I hope that this award would inspire more young girls in Malaysia to do science and to choose science as a career. There may be voices around you telling you that girls are not good for science or are not good in STEM. Don't listen to these voices and just focus on your dream. Girl must dream big and work hard to realize your dream, because nothing is impossible. Again, on behalf of all the young girls and women scientists out there, I would like to thank MINDS for always stand up for supporting and promoting women in science. Thank you. A very good morning to all. First of all, I'd like to thank MINDS for the invitation. I really appreciate the platform given to share some of my words and thoughts. I'm a biotechnologist by training, and my research expertise is on food safety and microbiology. But today, my topic is not going to be focused on my research findings, but more on sharing my thoughts and journey on integrating art in science. When we talk about art and science, we see these two as distinctly different disciplines that do not overlap each other, just like mixing oil and water. Many people compare science with art and make judgments such as science is more important, or without art, there will be no science and there will be no society. Just like the picture I showed on the screen right now, science tells us that our left hemisphere of the brain is often associated with languages, with logic, critical thinking, numbers, and reasoning, which are often described as important criteria to be good in science. While our right brain is responsible for intuitive and creativity, which is generally criteria for art. However, neuroscience research today reveal that the two sides of the brain actually collaborate to perform a broad variety of tasks, and that the two hemispheres communicate through the corpus callosum. In one research, it was found that the abilities in subjects such as mathematics are actually strongest when both half of the brain work and collaborate together. Yes, while there are differences between science and art, but both science and art share a lot of similarities, and both actually work best when they are integrated. Science and art were more intertwined in the very early days, in the 16th to 18th century. That is during the Renaissance era. So the screen here shows you the very well-known German artist, Albert Dürer. He has produced many masterpieces that had great influence on natural science, and has helped many to actually visualize the changing conceptions of the universe through his artwork on the northern and southern skies, shown as polar projections over here. He has also drawn probably the first known pictorial representation of the syphilitic man, so which well record the symptoms of syphilis in human. Another great scientist, as well as artist, he actually is well known as artist rather than scientist, but he's also a great scientist and engineer, as shown today during the Renaissance, which is Leonardo da Vinci. He clearly shows us that during the Renaissance, the study of art and science was not perceived as mutually exclusive, but as more as integrated and informing upon the others. Leonardo, in his art pieces, has demonstrated to all of us that the importance of curiosity, dedication, experimentation, good observation skill with accurate recording, imagination and creativity in doing arts and science. Leonardo da Vinci clearly showed how art is being used to enhance science, and how science can be beautiful and trigger our internal mind also. Yes, just like Charles Darwin once said, science and art are like two wings of a bird. Communities which use these two wings, they will fly and they will become free. Indeed, Science and art are the nature of human attempts to explore and understand the world we live in. If we integrate science and art together, instead of seeing science and art as opposing each other, so we will be able to discover more wonders, create more innovations, and push for advancement in science. Common features shared by both the disciplines include important skills such as observation, experimentation, problem solving, and openness to change. I feel really sad when I hear people saying that art is useless. And we actually see art being removed from our children's education when time is not enough or allowed for them to go through art. This is particularly shown during the online classes during the pandemic. Art is not being taught, but the focus is more on the main subject and also science. I think this is actually wrong. So in fact, art could actually help kids understand and comprehend science better. Science is a form of creation. So the purpose of science is to understand the world we live in and create within the world. So those innovation, those new technology that we see need creativity, both imagination and creativity. The reason why art is necessary to science is because creativity actually involves imagination, and imagination needs visualization. So things we are able to conceptualize, visualize, and imagine in our mind are things that we actually can create. So if we have the right tools to do so, which is science and the other technology. So therefore last year, YSN ASM actually came up with this idea to organize the Junior Science Artist Competition with an aim to release the imagination and creativity of children in science. We asked the kids to draw the world they imagine after the pandemic and how science could actually help in their life. So we get very interesting art pieces and even creative design and innovation. So as you can clearly see over here, these are all the art pieces created by kids. So we believe that introducing art as a way to study science would allow students to understand that the core of science is actually creativity and the ability to visualize and imagine certain processes is actually very important to solving scientific problems. So therefore, science and arts are very, very much integrated. So you need to use both part of your brain to do so or to be a good scientist. So since small, I actually love drawing more than studying. I spend a lot of my time observing and drawing what I actually see. Nobody in my family would imagine that one day I would become a scientist. But yes, my curiosity to death and also lives, and diseases, after my beloved grandmother fall sick with Parkinson, followed by my father fall ill due to acute kidney failure when I was only 6 years old, has changed my path in life from wanted to become an artist or musician to scientist. Some say that I have shown great potential in art as I could draw very well. And I really, really enjoy doing artwork. I could sit there for hours, without moving, but just drawing. And it is a waste that I do not continue doing arts. So that's what my art teacher said, it's a waste. But I think that the skills I learned in art actually helped me to understand and comprehend science concept more easily and better. So nothing is a waste. And I continue to explore art in science, and I see a lot of art elements in science. And I do also see how science could be applied in understanding art better. So let me just share very quickly, a little bit on how art has inspired my research. So my research is on food, and in food, the way of cooking and careful formulation of food ingredients, such as by adding different type of herbs and flavors, be it artificial flavor or natural flavor. It is an orchestra of aroma that will actually increase appetite of consumers. It is a field of science and also art. Food aroma and odor plays an important role in food sensation and also intake. Food aroma and odor also contribute to the grading of wine, cheese, and fermented food. Of course, up until today, we are still using human tester, like human nose, in sensing, smelling the aroma and giving a grade, like to the maturation of cheese and the other type of food. So during my PhD work, I was super curious about the smell produced by different microorganisms. I work with microorganisms, a lot of microorganisms in food. We humans can smell spoilage microorganisms in food, but we cannot smell the presence of foodborne pathogen. That is what makes foodborne pathogen undetectable and dangerous in food. So imagine if foodborne pathogen could be smelled or could produce a type of smell that we can detect, just like spoilage microorganism, then there will be no, there will be lower, way lower, like cases of foodborne diseases or food poisoning cases. But it seems like that we can't smell and differentiate food that has been contaminated with foodborne pathogen. But it seems like some of the animals could actually differentiate, like food that has been contaminated, and or even like human with certain bacterial infection. So although yes, nothing to do with my PhD work during that time, I was too curious that I went ahead, like to design a very small experiment to detect the volatile compounds produced by foodborne pathogen in chicken. So I compare the chicken that has been contaminated with Campylobacter and no Campylobacter. And I found very, very interesting findings that time that lead to my continued research on this field. So the research, like we found that there are different microorganisms present in food, and these different microorganisms produce different volatile compounds. But often, like these foodborne pathogen, particularly the disease-causing bacteria in food, often masked by large amount of other volatile compounds, which means that the foodborne pathogen usually do not produce high quantity, high amount of distinctive smell that is detectable by human nose. Without a sensitive nose, we cannot tell the difference between like the volatile compounds that has been produced by the foodborne pathogen. So this project is still ongoing, and we continue to profile the different disease-causing microorganisms in different type of food, hoping that in the future the findings from this work would produce an electronic sensor that will allow us to detect foodborne pathogen in food more rapidly and in real-time manner. So finally, so I would like to just end my talks by stressing that science lacks the art of imagination and creativity. There will be no new breakthroughs and advancements in science. Let us unite science and art to soar to new heights. With that, thank you.
Design thinking, computational thinking, examples of transfer, and flavors of reuse from e-learning, e-business to e-health by Professor Dr. Lee Chen Sing from Sunway University. Professor Lee is an IEEE senior member since 2007. She won her Fulbright Fellowship at Lehigh University, working on artificial intelligence, computing, and information system. And she also won her 2009 10 Outstanding Young Malaysia Award on Science and Technology Development by the Junior Chamber International Malaysia. She has spent 2 years at the Graduate Institute of Network Learning Technologies at the National Center University, Taiwan, on the teaching of mathematics to students in grade 1 to 3. And the next 2 years, she spent her adventure in the Faculty of Creative Industry, University Tunku Abdul Rahman, Utah, under the leadership as when she was in MMU. And this has provided invaluable insight into another side of design-based research in games design, preschool and graphic design. Now she has returned to computing and information system at Somewhere University, building mainly on computing fundamentals in the past. So let's welcome Professor Dr. Lee. Thank you.
Good afternoon, Tan Sri Ong, President of MINDS, Dato Dr. Chu, Vice President of MINDS and Chair of LadyMINDS, MINDS Committee, MINDS Secretariat. Ladies and gentlemen, thank you very much for giving me this very invaluable opportunity to share on design thinking, and also competition thinking with regards to flavors of reuse, transfer from e-learning to e-business to e-health. I'm Chen Sing and I am from Somewhere University, but also representing the Fulbright Alumni Association of Malaysia, Multimedia University, and also University Tunku Abdul Rahman due to the contents within. The discussion will be with regards to the SDGs 3, 4, and also 11. SDG 3 is well-being, which correlates to e-health. SDG 4, equal access to quality education, which correlates to e-learning. And SDG 11, smart cities and smart communities, which correlates to e-business. So the key components will be design thinking, computational thinking, and also interdisciplinary synergies. The design thinking process is very similar to the agile methodology process as well as a joint application development process. It emphasizes a lot on empathy, which requires the use of multiple instruments in order to better understand the context of the users themselves, and also their challenges, pain points, and how they make decisions. So most importantly, it is to optimize. So with multiple and iterative joint application development process based on incremental mockups, then it should result in higher satisfaction and also better performance and higher productivity. These are some examples by EDO University. So the first one is project blocks. Here, the team actually wanted to help kids learn to code. So they found out that the kids are actually very familiar with Scratch as well as Lego. Therefore, they came out with small decomposed building blocks whereby the kids will be able to assemble any form of algorithm. Moonrise is to help those who are financially challenged in financially challenged times such as now, and they apply the concept of grant. With regards to computational thinking, we are more familiar with the definitions which are provided by Professor Jeanette Wing in 2006. They are decomposition, abstraction, pattern recognition, and also algorithm design. So let's look at some examples. The first one is these two robots. So in terms of pattern recognition, we can say that these two robots actually share the decomposed heads, decomposed bodies, and also decomposed legs. And of course, their hands or their fingers are slightly different because of the purposes that they serve. So in terms of algorithm, also it will be dependent on the purposes that they serve. So say, for example, this particular robot is wearing the dark sunglasses. So he is most likely a dancer, whereas this very cheeky and smiley robot, she looks very friendly, so she can be a receptionist or she can be a waitress. So the algorithms can be pretty similar to a certain extent. With regards to abstraction, we have 4 or 5 of these robots, and they are of different forms and also different functions. Then we will be able to say that, oh, okay, this is the commonalities, and based on those commonalities, then we can actually fit into the libraries and they can be reused and also adapted or transformed by some other designers. When we look at this clothes hanger, that is also pattern recognition, because if we look closely, then this is the head, this is body, and this is the leg. And incidentally, the legs over here has rollers. So whatever applies to the robots in terms of moving from point A to point B can also apply to the clothes hanger over here. And whatever else applies to this, as well as is in the library, can also be applied to this smart clothes hanger. So that is going to be a very interesting smart clothes hanger. Coming to the younger generation, even younger generation, then we have Professor Brennan and Resnick's concepts, practice, and also perspectives definition of computational thinking. Professor Resnick is the founder of Scratch, and therefore he is always looking towards very, very, very young kids. And he wanted, or rather they wanted as much as possible to encourage the younger generation to try to improve their local community as well as to eventually improve the world. So they say that it is very important to identify the problem and also to hypothesize. And with this internal motivation, then they will be able to express themselves in order to seek more optimal solutions and also to connect with others. Computational concepts, as you can see from these 7 main concepts over here, they are actually pretty much algorithm design. Computational practices over here is a combination of the algorithm design, decomposition, and also abstraction, as well as pattern recognition. But over here, you see also reusing and remixing, which is actually refactoring. Some examples of computational thinking. This is the common Scratch that we know, and this is the part which is less known, which is the fractal part. So the fractal over here are actually pattern recognition in computational thinking. So different kinds of fractals that you see over here, and this is the fractal in real life. This is the antenna design, which is also based on fractals. And this is another example of buildings which can take different forms because of projection. And of course, this is very interesting if we look at the way that users can interact with the buildings. And of course, fractals can also be used in economics and also in biology. Let me share some examples of my past research in MMU from 2000 to 2005. So this is on the Onto-ID authoring tool, and then there is also the Malay Agent Assisted Collaborative Concept Map, and this is Onto DNA to accommodate external resources, and there is this Onto this visualization tool, which is to enable portability between XML and also RDF, because at that time it was the semantic web era. And to bridge everything is the open and shareable student model. This is a screenshot of Onto ID. This is Merlin, so this is the chat box, and this is a hint from the knowledge base. This is Onto DNA, this is Onto this, and this is my open and shareable student model. I was also very fortunate to be able to go to Lehigh University and Georgia Tech in 2009 on a Fulbright Visiting Scholar Fellowship. So in the Lehigh University, I was very fortunate to work with Professor David Blank to work on the Lehigh Valley STEM education project, and this is what I came up with. This is actually template based, and therefore this concepts actually. So this is a goal setting, this is the standards part, and then this is the analysis of the learner's needs in terms of the prior knowledge as well as the future target knowledge, and then the learner's environment and differentiation. This will be in terms of the evaluation. I was also very fortunate to be able to collaborate with Professor Janet Kolodner. At that time, in 2009, it was ex-President Barack Obama's first inauguration, and he proposed sustainable development as the way forward for the United States. So Malaysia and I think many parts of the world also followed. And therefore, we were trying to do a reference modeling in terms of the sustainable development curriculum between the US and also Malaysia, and we were working on strategies and tactics. This remains also a concept. So in 2005, or rather from 2013 to 2015, I was in University Abdul Rahman, and I was very fortunate to work with the graphic design and also the pre-service preschool teachers. So here, these two examples are on sustainable fashion. So the student actually labeled it as travel through time because he or she wanted to emphasize on the decomposed part, whereby different materials can be felt, but if we are not careful, then we may not even have these materials anymore. In order to lead the user to more information, there is a QR code over here. And here we see two different examples. This is actually a mystery box, so the kid will be able to open, and then there will be a lot of different artifacts which the kid will be able to assemble. Let's also sing along, and this is a crafted physics project made from reusable materials. This is another example which is adapted from Stanford's wallet project, which is a design thinking project, but I converted it into e-wallet because the top management at that time wanted to experiment with interdisciplinary research. So the graphic design students say that what we need is basically to analogize the wallet with the handphone, the tablet, or the laptop. So they say that the unlock function can be sensor-based in order to detect the face and also finger and also the thumbprint. And since everything is digital, then we do not need to have physical cards. And therefore, all that we need is actually just a touchscreen. But of course, we want things to be multi-purpose, and therefore the GPS should be in. So in terms of aesthetics, being graphic design students, they're very enthusiastic about color changes and also day and night changes, illustration changes. And they say that in order to add in the effect, then we should also enable swivel, and the particular wallet must be pretty lightweight. So in order to charge, then they say that very easy one, the particular wallet, okay, e-wallet, okay, can be kept in our pocket, and as we walk, then it should be able to charge by itself, so we don't need to bring anything extra. So it's really going to be very lightweight. In Southern University, there is again the theme of sustainable development and also sustainable design. So over here, the students, 3 students actually, they suggested that we can actually come up with a system to encourage people to recycle their clothes and also to be able to see others uploads of clothes. So all these uploads will enable supposedly people to buy if they are interested, because some recycled clothes are not cheap and they are actually in pretty good condition. And there may be designers who want to combine the designs, and there may also be manufacturers who want to manufacture all these designs by the designers. So it is multi-pronged and also multi-stakeholder. This one is to enable people to do interior design. This is an example of role-playing game. So depending on the choices made by the users, then they are able to look at different places of interest and also to be able to zoom in. And this one is tile-based, similar to Reddit's pixel art, whereby these particular tiles will be can be colored, and of course people can also share memories. So in terms of e-health, I learned this from University Tunku Abdul Rahman. These are actually 4, 1, 2, 3, 4, and I use this for the Alzheimer's Disease Foundation of Malaysia seniors, and also for Information Systems Analysis and Design class. So this looks different from this because I practice empathy, and therefore I asked the nurses over there what kinds of shapes the seniors are familiar with, and they said this kind of shapes. So okay, so I changed the shapes, so these kinds of shapes, and then ask them to arrange just like, you know, the Lego blocks and also the project blocks. And then from this, I was able to identify a bit of what their feelings and thoughts are, as well as their cognitive level in terms of their ability to manipulate objects. So it was very interesting. Then here, this is specially curated to help the seniors to prevent falls. And this is data mining, a collaboration with Salesforce to identify the clicks and interests of the seniors, which will then determine the content over here. This one is more towards finding objects, hidden objects actually. And this is actually to test schema. Is part of a cognitive experiment actually. There's another version of it, which is black and white. And this is object-oriented kind of Monopoly game. So in general, what are our findings? We find that the design factors among the better performing students for both the creative industries and engineering sciences are actually very similar. So this is very craft-based, although there are links to the computing, and this is very web-based, but we also find that the design factors are actually very similar to the design factors over here for the Scratch Fractal projects, and that is the better performing students are able to traverse between global and local, and their logic is very good, and then they are also very multi-sensorial in terms of multiple channels, and they are very, very aware of the objectives of the audiences, and also to whether the audiences understand what they are trying to do. So if we combine these two, then we will look at this refactoring, which is similar to Professor Brennan and Resnick's remixing and also reusing. And therefore, the weights that we apply and what do we apply to refactoring is the most optimal. So I would like to thank Multimedia Unici and also my ex-PhD supervisor, Professor Yashwan Prasad Singh, mostly for its IRPA and also eScience grant, JICA, Fulbright Commission, Lehigh Unici, especially Professor Glenn David Blank and also Professor Chua Mui Chu, Georgia Tech, Professor Janet Kolotner, and also Professor Ashok K. Goel. Professor Ashok was very helpful and nice. I learned a lot about structured behavior function from him. So SBF is very similar to computational thinking. I would also like to thank University Tunku Abdul Rahman's top management at that time when I was a faculty, because they are actually the same top management as when I was in Multimedia Unici for 12 years. So 12 plus 2 is 14 years. So thank you very much also to Minds for many opportunities to learn about internationalization as well as technology and innovation. Thanks to Dr. K. Daniel Wong for collaborating with me in Unici Tunku Abdul Rahman and also Sambal Unici. Thank you to Mohi and Moi for the FRGS grant, Dr. John H. Hughes for his kind input on neuroscience, and Professor Bojang, my collaborator for the Scratch Fractal project, Professor David E. Drew for his collaboration in the ICCE, and also my ex-colleagues in the Graduate Network Learning Institute in Taiwan when I was there for a very short period of time. Thank you very much.
Think Beyond Numbers by Professor Dr. Noor Aisha Hamza. Her area of expertise is exploratory data analysis, robust statistical modeling, and computations. She holds with her more than 25 years of experience as an academic with the Institute of Mathematical Sciences, University Malaya. Throughout her career span, she has been in various leadership positions as the head of department, Deputy Dean of the Faculty of Science, Deputy Director of Global Planning and Strategy Center, and Director of Global Planning and Strategy Center. Professor Dr. Noor Aisha has contributed articles to numerous journals, book chapters, research projects, and postgrad supervisions to her credits. Let's welcome Professor Dr. Noor Aisha. Thank you.
Thank you for the kind introduction, Professor Dr. Leong. Hello and good afternoon to everyone. First and foremost, I would like to thank Tan Sri Emeritus Professor Dr. Augustine Ong, the President of the Malaysian Invention and Design Society, MINDS, for inviting me as a speaker in the field of mathematical science to this session. My special thanks also goes to Dr. Chu Yuan Mei, the Vice President of MINDS and the Organizing Committee. I'm Aisha and I'm from the Institute of Mathematical Sciences, Faculty of Science, University of Malaya. I'm also representing UM Center of Data Analytics. The title of my talk is Think Beyond Numbers, Reasoning and Creativity. In this talk, I will not be presenting an academic research work, but rather to share on the prospect and outlook in mathematics. In this talk, I will provide an overview of mathematics and its importance, career opportunities. I will briefly touch on emerging roles for mathematics and ends with some practical experiences. Mathematics is an area of knowledge which includes the study of topics such as numbers, algebra, geometry, calculus analysis, right through to probability and statistics. Most of the activities in these areas consist of discovering and proving properties of abstract objects. Applied mathematics and pure mathematics are two different but closely related branches of mathematics. In applied mathematics, we deal with real-world problems and phenomena, trying to model them through mathematical equations and formulas in order to better understand and manage them more efficiently. Also, the important connection with other disciplines that may inspire interesting and useful mathematics, where innovative mathematical reasoning may lead to new insights and applications. Modern areas of applied mathematics includes mathematical physics, mathematical biology, control theory, aerospace engineering, finance, computer science, and many more. Pure mathematics, on the other hand, deals with abstract entities, finding relations, patterns, and structures, and generalize them whenever possible. With the rise of the modern science and technology era, the need for mathematics started to change. Today, we are witnessing the importance of many areas in pure mathematics with exciting current research direction in most of the major fields. Amongst them are linear algebra, widely used in image recognitions, text analysis, and also dimensionality reductions, calculus in optimization techniques, mathematical modeling of artificial neural networks for performance improvements, group theory, which is very abstract, now has numerous applications in chemistry, physics, and mathematics itself, graph theory in chemistry and computer science, and complex numbers being widely used in electrical engineering. Other important areas also include algebraic topology in data mining, number theory in security systems and cryptography, Turing theory of computability. This provides the theoretical basis to computing, wavelet and Fourier transforms, which is widely used in computer graphic designs. Statistics is another area where statistical knowledge helps us in using the proper methods for data collections, employing the correct analysis, and effectively presenting the results. Statistics is a crucial process behind how we make discoveries in science, decisions based on data, and in making predictions. Now, all in all, understanding the world through math gives us a way to understand patterns, to quantify relationships, and to predict the future. Some important mathematics-related skills includes reasoning, critical thinking, creativity, problem solving, and innovation. Reasoning is a process of manipulating and analyzing objects, representations, diagrams, symbols, or statements to draw conclusions based on evidence or assumptions. This is a skill in integrating mathematics knowledge into real life and everyday problems and using mathematics to conceptualize complex problems. Critical thinking, creativity, and problem solving supports the development of the ability to analyze a given problem from different perspectives, structure, organize, and evaluate information, generate multiple ideas, and to choose a proper method to deal with unfamiliar mathematical situations. As for innovations, living in a data-driven world, turning data into information and intelligence can generate business value. Companies are relying more and more on data to guide their decisions, and employees with such skill sets and able to translate into actionable decisions are extremely valuable. Career opportunities in mathematics. Most mathematics and statistics programs have at least 40% of core compulsory courses comprising of calculus, linear algebra, mathematical models, probability and statistics, programming at the intermediate at the very least. The remaining 60% will focus on the program pages. The logical thinking, problem solving, and decision-making skills learned from mathematics program are highly valued by employers across many job sectors. Among the career opportunities for graduates in mathematics and statistics are in business intelligence. We have statisticians, data analysts, data scientists, mathematicians. In system security, we have cryptographer, programmers. In financial sectors, we have financial analyst, actuary, investment manager, investment banker. And in education sector, we have teachers, lecturer, and researchers. Now, in the top 10 skills of 2025, as quoted from the World Economic Forum 2020, 50% of these skills are related to problem solving skills, 20% are related to technology use and development, while self-management and working with people skills makes up 20% and 10% respectively. 70% of these are mathematics-related skills would have been picked up at the degree programs. As for the emerging skills, as identified as being in high demand within organizations in Malaysia, these are very similar to that of the top 10 skills of 2025. Now, following the list of emerging skills of 2025 needed in Malaysia, here's the Future of Jobs Report 2020 from the World Economic Forum for Malaysia. Emerging jobs in Malaysia are in business intelligence, data science related, big data, and cybersecurity specialists. In an economy in which automations and AI is threatening so many careers, the demand for mathematicians will continue to grow until at least 2026. Now, looking at the skills required for data science and data analytics, most mathematicians graduate would have already acquired the overlapping skills. In fact, many mathematics graduates would also have acquired other skills such as algorithm development, machine learning, data analysis software, and data visualizations from their electives. The required mathematics-related skills allow graduates in mathematics and statistics to be easily trained and absorbed as data scientists and data analysts. Now, a quick look at the Occupational Outlook Handbook of the US Bureau of Labor Statistics. The overall employment of mathematics and statisticians is projected to grow as much as 33% from 2020 to 2030, much faster than the average for all occupations. For Malaysia, the prospects, I believe, is similar. New emerging roles for mathematics. Mathematics can play supporting roles and leading roles in practice. Supporting roles involves employing or applying existing sophisticated mathematics, whereas the lead roles involve active research such as research in computational mathematics and statistics, bioinformatics, and optimizations. Now, some examples on these roles can be found in business intelligence and analytics, data science, and healthcare. Now, in business intelligence and analytics, statistics and data analysis, including data visualizations, are used to understand current market trends, predict future trends and opportunities, as well as in managing risk. Now, altogether, these forms important components in business intelligence. Having these abilities will certainly help enterprise to boost their ROI through insights about their business, customers, markets, and products. According to AccuVet 2019, 8 emerging business trends in 2020 and beyond are in augmented analytics, augmented data management, data discovery, focus on generating actionable insights, nurturing a data-driven culture, conversational business intelligence, such as chatbots, diversified data job, and agile business intelligence development. Another important segment where statistics roles are crucial are in data science. Now, mathematics and statistics are the two most important concepts of data science. Data science revolves around these two fields and draws their concepts to operate on the data. Now, there are two main components of mathematics that contributes to data science, namely linear algebra and calculus. Now, while linear algebra are widely used in image recognitions, text analysis, and dimensionality reductions, calculus are used essentially in optimization techniques. Statistics, on the other hand, helps the industries to take careful data-driven decisions by developing a profound comprehension of data through data summarizations and inference. Now, in addition to these, analytical thinking and problem solving are the two most important requirements for any data science positions. In healthcare, data science has revolutionized healthcare and medical industry in large ways. Data-driven decision opens up new possibilities to boost healthcare quality. In healthcare, data science are used for medical imaging, genomics, as well as drug discovery, Also, data science are used to provide practical insights in building a comprehensive view of patients, consumers, and clinicians. And another usage of data science is to effectively handle, evaluate, and interpret big data by creating new paths in comprehensive medical care. Now, there are, of course, many more other examples of applications on the roles of mathematics in statistics. Now, I would like to share some experiences in mathematical modeling of COVID-19. It has been over 2 years since the start of the world pandemic caused by COVID-19. As of February 28, 2022, there is a total of 434 million confirmed cases of COVID-19 worldwide, including 5.94 million deaths. Now, in Malaysia, we have a total of 3.4 million confirmed cases with more than 32,500 deaths for the same date. For a total population of 33 million, we are experiencing 1 in 10 confirmed cases of COVID among the population, which is indeed alarming. Now, 79.6% of our total adult population has received 2 doses of vaccines. Now, although this is a high percentage, Malaysia is still suffering from a high number of COVID-19 infection and worsens with the emergence of Omicron. Now, in response to this problem, what we did back at ISM in 2021 was to investigate the most probable spreading patterns of COVID-19 in Malaysia. This study was led by Dr. Hifzuddin and computation performed by Khairul Anwar. Now, our study involves modeling and predicting the potential spread of the disease. Now, the SEIR model, a short form for Susceptible Exposed Infection Recovered model, represents one of the most implemented mathematical models for characterizing the epidemic dynamics and to predict possible contagion, also used for forecasting the progressions of COVID-19 epidemic. Now, the output from this will also be able to assist health authorities to predict possible infection risk in the population and for decision makers to better manage the resources and allocations needed in future. Now, the basic SEIR model uses 4 compartments, okay? That is, S for susceptible, E exposed to the disease, I infected by the disease, and R for recovery. Now, what we did was that in our model, we extend this basic model to include another 5 compartments. That is by compartmentalizing those with and without vaccines, and also to include another compartment for death. Now, compartmenting vaccinated and unvaccinated is useful because of the resources needed to treat those with severe symptoms varies from those with milder symptoms. We know that those without vaccination tends to have severe symptoms once infected compared to those who have received a complete vaccine doses. Now, all in all, this SEIR model helps to forecast how a disease will evolve by characterizing how people evolve during these times. Now, in terms of data, this model essentially uses 4 types of data for forecasting purposes. Okay, and these are the number of cases, hospitalizations, and death, the population size, disease characteristics such as transmissions and death rates, as well as intervention impact estimates. Some results based on the data from April the 1st through to August the 15th, 2021, with some assumptions. Our results shows that vaccination program has significant effects on the spread of the disease, with a downward trend as more people being vaccinated. Now, the immediate drop in COVID-19 cases is mostly due to mass and rapid vaccinations. Now, our results also suggest that lockdown measure was not properly implemented. Partially enforced lockdown would certainly lead to periodic fluctuations in the number of infections cases. Many studies have actually shown that lockdown is the most effective non-pharmaceutical intervention but this certainly requires strict regulations. Now, the result from these studies also suggests that COVID-19 vaccines efficacy can keep people from getting the virus. Now, although vaccine breakthroughs is still possible, vaccinated people have the antibodies which could prevent excessive downstream transmissions. Even if they are infected, they are less likely to get severe symptoms. Okay. Now, moving on, from our mathematical model, we also found that the inter-regional movement would lead to an outbreak. Now, those coming from states with a high vaccination rate seems to have no significant impact from these inter-regional movements, but not so for those states with low vaccination rates. Okay, and again, depending on the travel rates itself. Okay. Now, also we found that COVID vaccines become less effective at preventing infections or severe illness over time. Hence, the recommended COVID booster shots for vaccines that have fast waning rate. Now, herd immunity can be reached when enough people, 80 to 90%, have been vaccinated against the disease. All this, the last three, comes from our mathematical simulations. Now, the details on the methodologies and findings can be referred to our technical paper on modeling the spread, the effect of vaccination program and interstate travel in the spread of COVID-19 in Malaysia for 2021. Now I'd like to share some experiences in modeling loss data. Now, the focus of this work is to model loss data of a unimodal type with heavy tail. Modeling insurance of loss data of a unimodal type with a heavy tail has been an interesting topic for actuaries. Distributions that can mimic the heavy tail of loss data are crucial to sufficiently provide a good estimate of the associated business risk level. Now, several heavy tail models have been discussed in the literature, and this includes the Pareto, log normal, Weibull, and Gamma models. Now, besides modeling claim losses in insurance, the applications of these heavy tail models is diverse, for instance, to model financial returns and file sizes on network servers, among many others. Now, the recent focus on modeling such heavy-tailed data has been directed towards composite models following a paper by Corey and Ananda in 2005. Now, these models are made by piecing together two weighted distributions at a specified threshold theta. Now, if we look at this graph, okay, the first part of this graph will be modeled by the first weighted distributions called the head up to a threshold, followed by weighted distributions, the second weighted distributions, which is called the tailed. Okay. Now, although this F is a legitimate PDF, it is not, in general, it is not continuous and smooth. Okay. Now, to account for this criteria, we need to include continuity and differentiability conditions. Now, however, we must make sure that, or we have to have these initial PDFs for the Fs to be continuous at the threshold theta in order for the conditions, continuity conditions and also differentiability conditions to hold. Okay. Now, in this model, okay, the A's are the mixing weights, okay, and they are also the model parameters. Now, by applying continuity conditions, we can express the weight parameter in terms of the other parameters, that is in F1 and F2, okay, and to ensure that this is a PDF, a legitimate PDF, it is necessary to make sure that the summation of the mixing weights here sums to 1. Okay. Now, the threshold theta here, okay, is also regarded as another, as one parameter, model parameter. Now, using differentiability conditions to ensure a smooth functions, okay, so we can now express theta as a function of the other parameters of the composite model, okay, that is these two, by solving these equations. Now, although a closed form of theta may not exist, okay, by trying to solve this, the equation can be solved by using any numerical methods. Results and significant contributions. The results are obtained from an extensive simulation study, as well as from applications to two well-known datasets related to insurance, namely the Danish fire insurance data and the allocated loss-adjusted expenses data. Our finding illustrates the superiority of composite viable models in modeling highly skewed data to other common models in the literature to date. We also provided a general rule to develop a composite model where the total number of parameters of the composite model equals to the sum of the parameters of the two distributions used. For the composite models, the mixing weights and the threshold theta are expressed in terms of the model parameters and hence does not increase the number of unknown parameters. Details of the methodologies and results may be found in our paper, Modeling Loss Data Using Composite Models. So finally, I leave you with a quote on numbers from Lutsky 2008. Thank you for listening to my talk. Thank you.
Hello, the next sharing is on When Nutraceuticals Meet Nanotechnology by Dr. Fu Juyan. Juyan is a pharmacist with a PhD in pharmaceutical science from the University of Strathclyde, United Kingdom. She has more than 10 years experience working with nano formulations as a senior research scientist at the Malaysian Palm Oil Board. She is currently serving the Product Development Division to deliver value additions to palm bioactive. Her research interests focus on the applications of drug delivery system for palm bioactive. She is currently leading the nanotocotrienols team to scale up and transfer of technology to industry partners for commercializations. She is an active member in the Control Release Society and currently the Vice President of Malaysian local chapters. Let's welcome Dr. Fu. Thank you.
A very good morning, ladies and gentlemen. Thanks to the Malaysian Invention and Design Society, Tan Sri Augustine Ong and Dato Dr. Chu Yuan Mei for the invitation to this prestigious conference, celebrating contributions to national development by Malaysian women scientists. I'm very honored today to share my research experience on the topic when nutraceuticals meet nanotechnology. These are two game-changing components, and I would like to share my research experience on this as well as the chemistry between the two. A little bit of background on myself. I am a pharmacist by training. During my studies, I got very interested in the way a drug travels in our body from the time that we consume until it's being distributed to the target tissues or target organs to exert its health benefits. It's even intriguing to know that with drug delivery system or pharmaceutical technologies, we are able to program how the compounds behave in our body in order to optimize its efficacy or efficiency. I was very lucky to be given the opportunity to further my studies in pharmaceutical technology under Yayasan Beasiswa MPOB. During that time, I started researching on palm phytonutrients and using pharmaceutical technology to enhance the beneficial effect of palm phytonutrients. Fast forward 10 years, I joined MPOB as a research officer. I'm very lucky to meet a group of colleagues where I can be exposed to different research fields and experiences and manage to expand my research areas into clinical studies, nutrition studies, and many more. Now, this is one person that has always been there supporting me along the journey. That is my dad. This 72-year-old man is one person that constantly reminds me to live the best of my time at any age. He is still hiking for more than 8 hours, and I would love to imagine that I would have a similar level of wellness when I'm in my 70s. But what worries me is when I look at the number of mortality rates due to serious health-related suffering. That is referring to non-communicable diseases or chronic diseases. In this projection published in Lancet, by 2060, that is when I'm in my 70s, the number of deaths due to NCDs will triple compared to what is being reported now. NCDs, or non-communicable diseases, is contributing to up to 68% of global mortality, which is the major cause of death worldwide. More than 40% of these deaths were premature, under the age of 70. And because non-communicable diseases are chronic diseases and need to be managed over a long period of time, the economic impact was estimated at 7 trillion USD. We are talking about 36 million deaths per year worldwide from non-communicable disease. That is 7 in every 10 deaths worldwide are caused by NCDs. And these deaths are mostly avoidable or preventable. When we talk about NCDs, they are referring to cardiovascular diseases, chronic respiratory diseases, cancer, diabetes, and mental health conditions. The risk factors that contribute to these chronic diseases include unhealthy eating, tobacco use, physical inactivity, and excess alcohol. In conclusion, poor choice of lifestyle. It is about time to rethink about our current practice of healthcare. Rather, most of us are currently practicing sick care, where we do damage control, that is, reactively managing sickness as a result of poor lifestyle choices. Shifting from sick care to health care, it actually means proactively creating true health and preventing disease. This can be done by practicing good diet choices and lifestyle choices to maintain wellness. To understand what are good lifestyle and diet choices, we must understand the underlying mechanism. What these lifestyle changes will contribute is reactive oxygen species, such as smoking, bad diet, radiation, and pollution. They increase the oxidative stress in our body. And chronic exposure to oxidative stress leads to protein and lipid oxidation, DNA damage, and most importantly, chronic inflammation. While acute inflammation is important to stimulate healing, chronic inflammation is a repetitive cycle because it is an ongoing stimulus that results in increased inflammation, which further stimulate the stimulus, and it is a repetitive cycle that prolongs the inflammation in our body. In fact, as published in the Time magazine, chronic inflammation is the secret killer because it is found to be the link for many chronic diseases, including cancer, neurological diseases, autoimmune diseases, cardiovascular diseases, and metabolic diseases such as diabetes. Speaking about lifestyle choices and dietary choices, nutraceuticals play a very important role. It is defined as substance that's considered a food or part of a food that provides medical or health benefits, helping to prevent a certain type of disease. In fact, the definition of nutraceuticals is very broad between food, and pharmaceuticals. Mainly, it targets three different type of groups. First is a group of healthy people seeking to preserve wellness. Products include probiotics or weight management, and vitamin and mineral supplements fall under this category. The second category of target group is people with common health problems. They are taking products to slow the progression of certain diseases like diabetes, dementia, or mainly age-related diseases. And the third group of people who can benefit from nutraceuticals are patients with special nutritional needs. And products such as specialized formulas or nutrition solutions for chronically ill patients falls under this category. Classification of nutraceuticals can be done using their source or the benefits that they can do. So, for example, dietary fibers, probiotics, and prebiotics are usually intended to improve gastrointestinal health. Healthy fats are usually recommended for cardiovascular diseases and improve immune system, whereas antioxidant vitamins or polyphenols are recommended for age-related diseases such as degenerative diseases and also metabolic diseases such as diabetes and cardiovascular disease. Now, when we look at the perspective of pharmaceutical technologies, these compounds are being categorized based on their physical property. And this is important because the better we understand their physical property, the better we can understand how they behave in a human body after consumption. Basically, they are being categorized depending on their solubility in water and also their permeability across the gastrointestinal membrane. Most of the compounds fall under category 2, which has low solubility but high permeability. In the body, a chemical goes through several steps for absorption before they can reach to their target organs. First is the dissolution. Solubility becomes very important here because compounds that has high solubility mix well with the gut fluids that helps them to be transferred to the intestine in a more homogeneous way. In the intestine, they cross the gut membrane to be absorbed into the bloodstream, and this is when permeability becomes important. Once in the bloodstream, that is the bioavailability of a product. When a product are available in the bloodstream, they can then be distributed to the target tissues or target organ where they can start exerting their function. Of course, during this process, they are also subjected to metabolism and excretion. As for fat-soluble compounds, there is one more additional limitations that we are facing here, because lipid compounds, because of the low solubility, they tend to clump and aggregate together in big droplets. So at this stage, we need the help of bile salts to emulsify these lipid droplets to make small micelles. This is these micelles that can travel across the epithelial cells in the intestine to be absorbed into the blood system. So bile salt is very important in this process, but the secretion of bile salts is being controlled by the food and the amount of food and the type of food that we are taking. So the challenge is if we take fat-soluble compounds or fat-soluble nutraceuticals, the absorption of these compounds are dependent on when we are taking it, how we are taking it, and what we are taking it together with. Nanocarriers provide a solution to these limitations because Nanocarriers are being designed to load the active ingredients into these carriers that can carry the active components to where they are supposed to be distributed. It's literally like a vehicle that is designed to specifically carry a certain active compounds and travel in the bloodstream to be delivered to the target tissues. To give a perspective of how small is nano, how small can a nano carrier be? If you imagine a single strand of human hair, a nano carrier is 1,000 times smaller than the diameter of a single strand human hair. So why is this small size in nanoparticles so powerful? Imagine trying to dissolve rock sugar and icing sugar. Icing sugar is having particle size so much smaller than the rock sugar. So when we are trying to dissolve it in water, the amount of surface area exposed to water is much higher in icing sugar. So the time needed to dissolve this icing sugar is much less compared to trying to dissolve a rock sugar. And because of this property, if we are trying to taste rock sugar, we will, compared to icing sugar, we will feel like the icing sugar is much more sweeter compared to tasting a rock sugar. So a change in size and change in the total surface area actually changes the physical properties and also chemical properties of a compound. Coming back to nanotechnology, if we are reducing the size of a cube from 1 centimeter down to 1 nanometer, the total surface area actually increased by 10 to the power of 7. Here I would like to share some of our research work carried out in MPOB using palm vitamin E tocotrienols for nanotechnology. Palm vitamin E is different from other source of vitamin E because it contains 20 to 30% of tocopherols and 70 to 80% of tocotrienols. Tocotrienols are unsaturated vitamin E, and because of this unsaturation, they have additional benefits of 40 times more potent antioxidant and also in anti-inflammatory properties. Advantage of using nanocarriers for palm vitamin E is enhance the absorption for oral consumption, enhancing the bioavailability. Being a fat-soluble compound, using nanotechnology helps to emulsify the tocotrienols and make it into smaller size without the need of bile salts. Whereas for skin penetration, nanoparticles were shown to penetrate better on the deeper into the skin layers. Here I'm showing the prototype for nanotocotrienols. It is a vesicular system with a lipid bilayer, where tocotrienols can sit within the lipid bilayer. We found that the encapsulation efficiency is up to 40%, and this prototype can be available in powder form or in a liquid suspension form. We also coat the outer layer of this nanocarrier with a hydrophilic polymer. The advantage of engineering these nanoparticles is we can design how we want the nanocarriers to be distributed in the body. For example, to increase the water solubility, we add hydrophilic polymer as a coat outside the vesicles. This will improve the circulation time in the body. And also, we quantified the size of these nanoparticles at around 200 nanometers. We can also engineer the size by changing some processing parameters and also changing the components. So nanocarriers can be very versatile depending on the application, the intended application. As a proof of concept, we conducted a preclinical rat study to see if nanocarriers can improve the oral bioavailability of tocotrienols. Here we have graphs of plasma concentration over time, where rats were given unformulated tocotrienols in black and nanotocotrienols in red. From the three graphs, we can see a significant increase in plasma concentration of tocotrienols. When we look at the total area under the curve over 24 hours, up to 4-fold increase in alpha tocopherol, 8-fold increase in gamma tocotrienol, and 13-fold increase in delta tocotrienol was observed. For efficacy study, we did some preclinical study on cancer models. This is to see if administration of nanotocotrienols helped to reduce the rate of tumor growth. In both epithelial xenograft models and breast cancer models, the rate of tumor growth was reduced compared to non-formulated tocotrienols or untreated group. In breast cancer models, the tumor volume is 12 times lower if rats were given targeted nanotocotrienols. And this suppression of tumor growth leads to prolonged survival of the animal models. Also, in luminescence melanoma cells, we did an experiment looking at the luminescence expressing cell lines. We can see here that with nanotocotrienol treatment, the size of tumor was decreasing over time, whereas for non-formulated tocotrienols, we can see a gradual increase in the size of tumor. In summary, our studies with tocotrienols validated the hypothesis that reducing the particle size of fat-soluble compounds helped to improve the absorption, oral bioavailability, and subsequently increase the efficacy of tocotrienols when tested in anti-cancer models. I believe that nanotechnology is at the early adoption stage, especially for nutraceuticals. A quick search in the market, we saw nanocurcumin, nanominerals, nanovitamins. The opportunities are tremendous. I hope my talk is able to inspire more young women scientists to join forces and towards the Sustainable Development Goals, that is to maintain good health and well-being. Particularly for NCD, the target is to reduce NCD by 1/3, and this requires strong R&D support and strong capacity to manage health risks. Before I end my presentation, the biggest lesson that I've learned, and I will always remind myself, is to seize the opportunity. And this requires a lot of courage and a strong support system. In MPOB, I am very lucky to receive strong support from the management. I would like to take this opportunity to thank the Director General, Dato Dr. Ahmad Parviz, Director of Product Development and Advisory Services, Ms. Rosida Razian. She is one woman that can always see beyond challenges. And to Dr. Kanga, the Head of Nutrition Unit, she's been a great mentor to me since I joined MPOB. Thanks to Dr. Chu, who has given me a lot of guidance and direction. And last but not least is to Dr. Kala, the former Director of Product Development and Advisory Services, who helped me shape my values and aspirations since I joined MPOB. And of course, to my family and children, who always keep me on my toes. With that, I end my presentation. Thank you and have a great day.
Thank you, Dr. Fu. Now we would like to invite all the speakers for the Q&A section. Let's welcome Professor Dr. Noor Aisha, Professor Dr. Lee Chen Sing, Associate Professor Dr. Chai Lai Ching and Dr. Fu Juyen.
Hi.
Welcome back. Very impressive sharing. Very impressed with your achievement as well. Now we received quite a lot of questions from the participants. The first questions will be to all the speaker here, what is your biggest challenge you have encountered? And second question, if you are given a chance, will you still keen to be a lady or woman? Thank you. Can we start with Professor Dr. Noor Aisha?
Right. Okay. Yeah, I think for me, mathematics doesn't sort of differentiate between male and females. Okay, it's a kind of general. Okay, and the probably biggest problem in mathematics is that to get some of the work across, okay, it needs a lot of collaborations, and that's a problem that sometimes we have, because trying to understand another sort of subjects which takes sometimes quite a lot of time. I think for mathematics, many of them, they are just like mathematicians, so it's kind of like trying to sort of learn something new from their sort of scope, okay, which probably take some time, but I think those related to physics and economics, maybe that's not too bad, okay. So yeah, that's a challenge for me. Yeah.
If you are given a chance, will you still keen to be a lady?
Oh, yeah, of course, yes.
Why?
Sorry?
Why? What is the reason?
For what? For what? Sorry.
You still want to be a lady?
Yeah, I think in terms of being a lady, I mean, because the subject itself is like, as I say, it doesn't sort of differentiate male and females, okay. And I think being a lady, is quite nice because I mean it doesn't differentiate, but I think for ladies, I think they have a lot of sort of patience in doing a lot of job, and they're very sort of meticulous in looking at their work. So I think that kind of things actually helps. So I just want to be a lady that, right?
Okay, I would like to invite Professor Dr. Lee to address these two questions as well. Dr. Lee?
Hi, thanks for the question. Am I coming through loud and clear?
Yes.
Okay, all right. So the biggest challenge that I have encountered, I think a lot of students, whether from the science or from the arts, they are very interested in the other side because it is very attractive. And whenever I try to ask them to, go into D, then they are like, oh yes, okay. So the capacity to learn is always huge, and is very encouraging. The only problem sometimes is that they get so attracted that you have to come back and try to really develop very strong base so that you will be able to not just be a jack of all trades, master of none, but you'll really be very, very good in what you are doing, and still be very open to other disciplines in order to enrich the discipline itself. So I think that is the biggest challenge. Our students are just too curious and very, very capable. Okay, with regards to the second question, if given a chance, will I still be a woman? Yes, although I do not face any difference, between men and women in the computing or in the creative industries. I think everybody should just be who they are meant to be. So I hope that answers the question. Thank you.
Okay, thank you. Let's welcome Dr. Chai.
Yeah, thank you, Prof. Leong. I think probably the most challenging just kidding, yeah. So probably let me share a little bit when we talk about the most challenging thing, for me at this moment as an emerging scientist or researchers in the field, what I feel is most challenging is is how to balance, like, you know, the different roles that we are playing at the moment. So particularly, like, you know, for women, even like, you know, myself, I am still single, so I do not face, like, you know, an issue of, like, you know, I need to spend a lot of time, like, you know, to man a growing family. So, but I do see, like, you know, a lot of my colleagues, like, you know, women colleagues, that they are struggling, like, you know, to have, like, you know, a balance of time, like, you know, to attend to their family and to their kids, like, you know, in which, like, you know, they are playing a role as a wife and as a mother. So, but at the same time, like, you know, as a young researchers, like, you know, scientists, so this is the time that you need to work on your career as well. So how to progress, like, you know, in your career, how to spend time, like, you know, in doing research. So that is something, like, you know, also, like, you know, kind of, like, you know, having a lot of, like, you know, women scientists having dilemma. So, but as a leader, like, you know, like myself, like, you know, need to play a role also, like, you know, in research management administration, as a YSN chair. So I think Prof. Leong faced the same issue also, how to split your time and to be balanced, like, you know, taking care of all of these, attending to your student, attending to your family, attending to all the different works that you need to do. So to promote science and advanced science. I think this is one of the most challenging thing, and I'm still learning on how to prioritize and how to arrange time. So I always believe that, like, you know, when you have a will, so there's always way that you can do it. But I don't think that there's a way, like, you know, that you have, like, you know, a lot of people talk about work-life balance, but integrating our work, like, you know, in life probably is the way forward, like, you know, rather than separating both. If I'm given a chance, like, you know, to choose, I think I would still want to be a woman. I think, like, you know, in this era, this time, I want to be a woman, because we are just very, very lucky, like, you know, to be born in this era whereby women social status, like, you know, are being being of concern, and there's been a lot of opportunity, like, you know, being given, and a lot of works, like, you know, have been done, like, you know, to improve, like, you know, the welfare and the condition that we are in right now. So at this moment, yes.
Thank you. Dr. Fu?
Thank you, Prof. Leong. Can you hear me clear?
Yes, can.
Yes. I think I share same feeling with Dr. Chai also, is on the balancing on the time management, and sometimes also on the self-identity, because we are playing so many roles, and in every roles we try to identify ourselves, what we want to achieve, and that is when we are switching between the roles, and it's a bit challenging to know what to achieve on that specific roles, especially, yeah, we have families as well. So I've sometimes I think allocation of a little bit of time to ourselves, some me time, that allows us to self-reflect, and that helps a lot in trying to find an anchor, and also and communication and managing expectations. I think that is some of the challenging things that I'm still trying to learn. Yes. And second question, yes, I'm still keen to be a woman, and the reason is I have enjoyed every bit of it, and I'm looking forward to the future. Thanks.
Thank you. Okay, question 2 address given by Ms. Sharifah from University Science Malaysia. Question to Dr. Chai Lai Cheng. So why it is so important to study microorganism in food?
Yeah, thank you for the question. So I think, as, like, you know, Prof. Neil also mentioned, like, you know, earlier on, like, you know, shared a lot about microorganism. When we talk about microorganism in this pandemic time, like, you know, to a lot of public, microorganism is something bad. So microorganism, bacteria, viruses, all causes disease. But actually it is not the case. So when I study about, like, you know, food microorganism, of course, like, you know, my field is in food safety. I always look into the bad, the nasty part, like, you know, of the microorganism in food. But over the time, like, you know, we sort of, like, you know, start to realize that it's very impossible, like, you know, to remove all the microorganism from our environment and particularly in food. And microorganisms play an important part. So when we look into microorganisms in food, we are not just looking into the nasty microorganism in food that cause diseases, but also the good microorganism in food that contribute very, very much, like, to food security and food sustainability. So in this study of food microorganism, so we do not look at food as, like, just the nasi goreng in front of you, or, like, nasi lemak in front of you, but, like, the whole food value chain, like, from the farm to the food that is being served in front of you. So the microorganism that play a role in the farm, like, in promoting growth, increasing yield and productivity, is as part of the very, very important study to ensure that we have enough food to consume throughout, like, that is part of the food sustainability. And throughout the way, like, how the spoilage microorganism come into the system that cause deterioration of food, and therefore less food will be able to be distributed to people. So how microorganism could be used, like, in improving the quality of food, so that food are nutritious, so that food can be kept for a long time, and so that availability and accessibility to all people, like, they will be able to get it. So all those are all why it is very, very important to look into the microorganism in food. So to make sure that we have safe and nutritious food and enough food for us to consume in the future.
Good, thank you. So question 3 from Miss Dina, UCSI. Question 2, Professor Dr. Noor Aisha. Competence with data is always identified as one of the key trends in driving change in the 21st century. So how should university to ensure that data literacy of all graduates, irrespective of their degree programs, is at an adequate level? Professor.
Thank you for the questions. Yeah, thank you for the questions, Prof. Jong and also Ms. Tina. Okay, now data literacy is an education expectation which is required for all, actually. Now, to ensure that data literacy is adequate, among the recommendations that we can make is, number one, to infuse data literacy into the curriculum, into the students' curriculum. So actually what this gives is that it gives an unrealized sort of opportunities for universities, okay, to make an impact on the current generations as they prepare to move forward into the workforce. That's number one. And number two, I think in order to make sure that it is also at adequate level, is to probably, in many universities, what they have done is that this data literacy is usually taken for granted, that, they're seen to be at adequate level, which may not be the case. So I think in order to ensure that this is at the adequate level, it has got to be assessed, okay, remediated, and also amplified. Okay, so I think maybe these are the two sort of recommendations we can give, okay, to ensure that this sort of level is there. Because I think they, as we move forward, I think in all sort of, lines or areas of research or at work, data literacy is going to be very, very important. And people with that kind of literacy, I think they have a better prospect in that sense. Thank you.
Thank you. Question 4 from Mr. Arshad, UKM. Question to Dr. Fu. What are the major challenges of nanotechnology in Malaysia?
Thank you for the question. I think the major challenges is the gap between the industry and the academics, because when we are having networking in all the sessions that with academics, we find we have a very excellent scientists working on nanotechnology. They managed to develop state-of-the-art delivery systems, and it's very mature. And we also have a industry who is very eager to adopt new technologies, and they are continuously searching for new technologies to be applied in this area. And the advantage is nanotechnology at this moment. I believe it is at the early adoption stage, and it's going towards a mass adoption where more products will be coming out and the technology is maturing itself. So I'll be looking forward for more products, especially consumer products that are using nanotechnology.
Good. So now that's all. No more questions? Or one more. Okay, question 5 from Ng Kim Yong, a private consultant, to Professor Dr. Lee. In your opinion, how to encourage female students to do science, especially in ICT in Malaysia? So what are the challenges that you have faced and how to go about them in this field, which is mainly dominated by male researchers or scientists? Dr. Lee?
Hi, thank you very much, Dr. Yong. Okay, just now I had problems with unmuting. Okay, so thank you very much again for the question. Okay, in your opinion, how to encourage female students to do science, especially in ICT? From all my years of teaching, there's almost 25 years. There are actually a lot of female students doing ICT in Malaysia, but of course, we should encourage more female students. So the main factors which usually encourage any student to go to any course will usually be the interest as well as aptitude, salary, and also potential for growth in that specific industry. So a lot of people would say that we can actually do some refactoring among all these 4 and also try to play with maybe the stakeholders based on quintuple helix. So currently we have the industry, academia, and also the government. And also now 2 additional stakeholders will be the civil community as well as the environment. So the ecosystem is playing a huge role in encouraging students to go into ICT, whether male or female. So I think that is the best motivator, because it is ultimately the ecosystem which will attract, besides what is intrinsic. So all these multi-factor, multi-level kinds of considerations need to be taken in. Okay, and then what are the challenges that you face and how to go above them? I think the challenges will be more towards encouraging students to be very, very well versed in their respective disciplines while being open to other disciplines. Because as I've mentioned earlier on to another question, sometimes students are very attracted. So say, for example, computing and psychology, computing and art, and computing and color theory. Okay, so I mean, these are things which I cover in my lectures and also in my courses. So sometimes they are very super attracted, and then they go very, very deep, and then after that they say that they want to know more. Then I say, yeah, you can know more, and definitely you're encouraged to know more, but you're also more encouraged to go very, very deep and to really master the body of knowledge which is per your discipline, because you need to be master of one while being open to other disciplines, so that you will be able to participate in translational research, transformative research, in a more meaningful manner. Okay, so I think the challenge will be to highlight to the students about the importance of being very, very deeply grounded in their own discipline while being open. And the other thing is also to mention to them maybe that the ecosystem has to be prepared for very interdisciplinary people, because at this moment the certifications and then also the jobs, they are still per discipline. So yeah, so in certain parts of the world they are very inter-D, but they mask it so that the IEEE part is very obvious, and then they take concepts or principles from other disciplines and then they incorporate, but then it is still IEEE. So it's very interesting, and they do very, very well. So okay, I hope I have answered that. Then the other one is to rise above this, which is a mainly dominated field by male researchers. I think maybe we can learn from the sports people. They will usually be very, very focused. So say, for example, we take swimming. There are different competitors in different lanes, but at the same time, we need to set our target very clearly, and then we just focus on that particular target while being aware of our competitors. But the male researchers are actually our friends, our collaborators. So as mentioned in the previous answer also, I've never felt any difference between male and female, and to me, everybody is just who they are, and everybody is very unique and very special. So, and I don't feel much differences. So in that sense, I think we just need to try to become the best that we can.
Yeah, good. Last question, question 6 from Cheng Ching, Singapore Institute of Technology to Prof. Lee. How do you instill interest in computational thinking to students in the creative arts or studies? How can computational thinking be applied across disciplines?
Oh, okay. All right. So this computational thinking is actually part of the creative arts, because computational thinking, focuses a lot on pattern recognition, decomposition. So supposing we talk about decomposition, one of the examples which I showed just now was on the 4 shapes. That means to say the circle, and then I have the rectangle, and also triangle, and also the line. So the line itself is the nuclei, so to speak. Okay, so in this sense, when we think about how to utilize, how to assemble atomic levels, or nano levels, okay, and then at different levels of abstraction, we actually have different combinations. So say, for example, the example which I gave on the e-wallet, the Stanford Design Thinking e-wallet challenge, okay, so that was when they actually identified the problem, okay, into 4, and then after that, they were able to identify what do they want for each particular 4. And then they were also able to analogize. So that was the pattern recognition part. So actually, computational thinking is more natural in the creative arts and industries than in the science. If we think of it from a different lens. Okay, so how we assemble based on craft is the bridge between arts and also science. So I think, to imbue interest, we should always celebrate each discipline for what it is and what it can be. So when I tried to imbue interest in computational thinking as in the science aspect, then I was trying to, as much as possible, ask them, okay, so you link to, your videos, and then you link to your, say, for example, your friends or your social media, because at that time it was 2013 to 2015, right? So it was many years ago. So the technology was not so advanced yet, and now we are all going to IoT sensors and etc. etc. So that was how I tried to help them to think beyond their computational thinking in terms of their own disciplines to computational thinking across disciplines. Okay, so how can computational thinking be applied across disciplines? Okay, so I think I have answered this question. The best example was the Stanford e-wallet challenge.
Very good. Thanks all the speakers. Very, very impressive. Now we would like to invite Dato Chu to deliver her closing remarks. Thank you. See you.
Ladies and gentlemen, we have come to the end of an exciting scientific journey powered exclusively by women. Now, if there's one clear takeaway message from the conference today, it is that the country must tap into the enormous talent of women for national development. Professor Ngiao, the recipient of the Outstanding Woman Scientist Lifetime Scientific Research Achievement Award, show us the power of small creatures, the microbes. They can be beneficial to us, but can also wreak havoc, as COVID has shown us. She describes the importance of understanding microbes for better disease control. We must understand what happens when microbe meets human in order to devise appropriate preventive and treatment strategies. Dr. Fu Yuyan also showed that small is powerful, but this time in another dimension, nanotechnology. Nanoparticles can navigate and overcome biological barriers and are thus important delivery tools. There's one global shift towards natural derived products, such as nutraceuticals for health and wellness. However, bioavailability is often a limitation, and development of effective delivery systems is important. Dr. Fu described the application of nanotechnology to enhance nutraceutical efficacy using tocotrienol, a strong pump-based antioxidant, as a case study. Ladies and gentlemen, art can be a powerful medium for exploring the deeper meaning of science endeavors. Dr. Chai Lei-Chin, recipient of the Young Woman Scientist Award, who is a biotechnologist by training, talked about her journey of finding art in science. She shared with us how the arts have helped her learn and comprehend science better, and how she finds science in art and religion as well. Now, another person who described the blurring of the boundary between science and art is Dr. Sim Sun Liang, who viewed plant breeding as both a science and art. Art because you are designing a plant type best adapted to a particular environment, and there's good science going into creating it. Her years of perseverance and determination resulted in a pepper cultivar, semongkok, a musk, which is widely adopted by pepper growers in Sarawak. Now, for those interested in pursuing a career in plant breeding, modern technologies like CRISPR offer new exciting opportunities for breeding. Aquaculture is currently one of the fastest growing food production systems in the world. It is projected to be the prime source of seafood in the near future as wild capture fisheries approach the maximum limit. As Professor Dr. Irene Tan explained, now the fisheries in Malaysia need to continue along a sustainable path of economic development as capture fisheries have been pushed beyond their sustainable limit. Her team at USM has been carrying out research on oyster farming as a green sustainable form of aquaculture to improve the coastal environment and provide alternative source of income to traditional fishermen, thus promoting prosperity and protecting the planet. Prof. Noor Aisha described mathematics as more than just number crunching. Now, it contributes to the development of valuable skills in problem solving, creative and critical thinking, language, and more. It is the key to opportunity and contributes in fundamental ways to all facets of economy, from business, finance, health to defense. To participate in the world of the future, we must tap into the power of mathematics. Dr. Lee Chen Sing gave us a glimpse of digital disruption from the lens of the various sectors, from e-learning to e-business to e-health. She talked about Bloom's revised taxonomy, which focuses on outcome-based education, integrating technology in teaching and learning. In other words, technology becomes the frame in which education takes place. Ladies and gentlemen, a scientific discovery is only as good as its communication. Dr. Maha articulated the importance of communicating science. Science thrives when scientists communicate it effectively. However, scientists often struggle to communicate their work and discoveries to a non-technical audience. Now, the lack of ability to communicate science to a variety of audiences severely diminishes the potential of the work to positively affect society. Dr. Maha emphasized how engaging with various stakeholders supports research, development, and commercialization, as well as social, economic, and environmental well-being of a nation. Finally, ladies and gentlemen, now on behalf of Malaysian Invention and Design Society MINDS, I would like to thank our distinguished speakers who shared their valuable experiences in various facets of science. Special thanks to our sponsors, panel of judges of the awards, members of the media, participants, the conference organizing committee, and all those who have in one way or another helped this conference possible and successful. Your presence and support have been invaluable and gratefully appreciated. I also would like to express a very special thanks to Tan Sri Augustine Ong, President of MINDS, for his unwavering support for the role of women in science. To the students and future scientists who are among the participants today, I hope you got an overview of the possible career opportunities from our eminent scientists. My personal wish is for you to find your place in science. Once again, thank you very much, everybody. All the best and stay well. See you again in MINDS National Conference on Contributions to National Development by Malaysian Women Scientists and Inventors 2023. Thank you.